I think because Sabu sir is already traveling, so he's unable to connect. Hence, Dr. Shirley, would you like to? Sir, are you back? Sir, you're on mute, sir.
Yeah.
Yes, sir.
Yeah.
Okay,
Yeah.
now you're audible. Yes, sir.
Yes. Maybe if you permit, I will show you some slides. Can I?
Yes, sir.
Can you see my slide?
Yes, sir. We can see your slides.
Yeah. Dear colleagues, I'm going to talk about translation. I would say it's a missing link between research and practice. Can you see my slide, dear colleagues?
Yes, sir.
Dr. Nebu, can you see my slide?
Yes, sir.
Yes. As I mentioned earlier, it's a translation from bench to bedside. Taking our findings, our research findings from the laboratory into the clinic so that our patients are benefited. As I mentioned earlier, translation is a dynamic process, iterative process. It involves lots of work, synthesis, dissemination, exchange, and also ethically sound application knowledge. And translation always increases the relevance, applicability of your research finding. I would say if you can do a good translation, then you can say your research has a great impact. So impact of a research is always measured the amount of translation that you need. We have a beautiful slide here. Translation actually fills the gap between the research results and the research impact. When you do translation, we should ask certain questions. What should be translated? What should be transferred? To whom and by whom? These are some of the questions we always come across in the case of translation. I also wanted to show you a very important data. If you look at the work of Professor Wolf says that something like $1,000 billion per year is wasted in biomedical research, in health research, because of the implementation failure, because of the translation failure. So please remember the fact that all bench scale experiments cannot be translated. You will have a lot of failures. And I also wanted to remind you that only 14% of research findings reach the patient. If you have 100 patents, only 14 patents will reach the patient. That's again a good achievement. And I also want to show you another important statistics. It almost takes 17 years for research to reach practice. This is an average time period. So dear colleagues and friends, we have to be very patient when you do translation. When you look at the translation, there are different phases: T1, T2, T3, and T4. T1 is nothing but translation to humans. T2 is translation to clinical settings. And the T4 is translation to populations. So T3 is translation to practice, and T4 is translation to population. If you look at T1 in more detail, T1 has very important components of preclinical animal studies, human physiology. There are several steps involved in T1. Similarly, you look at T2, phase 3 clinical trials. So we have to follow all these protocols. Look at T3. T3 involves phase 4 clinical trials, health service research, clinical outcome research. And finally, if you look at the T4, translation to population, population level outcome studies, social determinants of health. So all these steps have to be followed very carefully when you do translation. That's why translation takes 14 to 15 years, sometimes 20 years. We also have to look at the ethical standard. That's very important when you do any translation studies, dear colleagues. Ethical standards have to be carefully followed. Translation research initiated in India. We have a School of International Design, Stanford, India. Biodesign is a flagship program of Department of Biotechnology. I request all of you to look into this institute details. Similarly, we have an autonomous institute of the Department of Biotechnology and the Ministry of Science and Technology for Translational Health Science and Technology Institute. Let me now show you what we are doing in Mahatma Gandhi University. Mahatma Gandhi University has a space. We have a Section 8 company, basically for translation. It's a small space, but we are making a bigger space under the support of Government of Kerala. And those who are really interested to do translation, you can approach us. We have a built-in space of 14,000 square feet. You are most welcome to visit our translation space. And some of the work that we are doing is, we are quite active in electrospinning and wound healing. We did a lot of work in this area. So we have to do several clinical studies to translate this into a real product, to translate this into a marketable product, particularly on tissue engineering. Let me now conclude. Dear colleagues and friends, the goal of translation research is to advance the project towards the next step in translation, preclinical research, clinical implementation, and public health stages. So I'm making a request to all my colleagues, let us work as a team. We have to have a lot of connectivity, we have to have a lot of collaborations. We want to translate our bench scale experiments into real products. And I wish you all the best and good success. You are most welcome to our campus to visit our MDU Innovation Foundation. Thank you very much.
Thank you, sir. Thank you very much for the enlightened speak. And I know that the MD University has got a startup company, a startup incubation center is there to initiate many translation research. So it is, I know, the focus of sir. So we'll be connecting to you, sir, after this meeting. So we got the clinicians and material scientists research. So there is a wide group of people has been joined.
Definitely, whoever is interested can visit our center.
Yes, thank you, sir. Thank you very much for your valuable time.
Thank you. Thank you.
Thank you. Thank you.
Now I hand over to Dr. Shirley. She'll be the next moderator for the next session. Dr. Shirley, you're now.
Thank you so much.
communication with the sponsor, all these day-to-day affairs will be managed by the site management organization, which will have its own set of clinical research coordinator and other research staff to help in organizing and managing the clinical trial. So these are the different players who are involved in clinical trial. So as I said, the sponsor will use a CRO to undertake a clinical trial, and the investigator will take the help of a SMO to conduct the clinical trial at the hospital site. Clinical trials are a very expensive affair. It could be as low as 1.3 to as high as 2.8 billion US dollars. So that is why it is only the big pharma, that is the large pharmaceutical companies, which have the capacity to manage uh, uh, the 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 risk involved. They should have a sufficient risk appetite to participate in a clinical trial, because it's not that every clinical trial that you do you're going to see success. At every stage of the clinical trial you will see almost a 30% attrition. Almost by the time a product comes to phase 3, you will find so many molecules would have already dropped out. So it is a high risk venture. So small pharmaceutical companies will not be able to handle that kind of risk, and that is why only large pharma companies are involved in the business of drug discovery programs, having their own molecules which they proceed and take it for development. But again, this is also changing of late, because if you see in 2020 when the pandemic struck, who are the people who are discovering new vaccines? It was not the large pharma companies. It was the small biotech companies which are involved in the in discovering those vaccines. Like for example, BioNTech, Pfizer vaccine was actually discovered by BioNTech. Similarly, Moderna vaccine also was a small biotech company was the one which initially was involved in the process of the discovery of Moderna vaccine, and subsequently Pfizer has taken over this company, and they have because in order to scale up the production and in order to manufacture at a large scale, we need the help of large pharma. So it's a very common practice for companies to be involved in drug discovery, small companies to be involved in drug discovery, but when they want to take it for development, then they would merge or collaborate with a large pharmaceutical company, and if it succeeds, it will be a kind of thing where that company is absorbed into the parent company that had collaborated with it. So, and the other aspect is the time. It takes a lot of time. It might take sometimes 15 years, even Dr. Sabu was mentioning this, even 20 years it might take up. So it's a long drawn out affair. It is not something that happens overnight. The pandemic was an exception. We saw things happening at a very rapid pace of 9 months, 10 months, 1 year, 1.5 years. But that is not the usual case. It usually takes easily 8 to 15 years. The reason is because for each stage, even the animal studies to complete each stage, and it's not that when you take a molecule, it works. Many times multiple failures happen, and then they have to go back to the drawing board, modify the structure of the compound to see how compound could be made better. And sometimes even failures happen at the clinical stage, at phase 1 you encounter safety issues, you might end up dropping the molecule altogether, or you might consider modifying the structure of the molecule and see if the modified compound has a better safety profile. So things of this kind will obviously take time, besides the actual time taken for doing the different studies. So it is a costly venture, and it is a time consuming venture, and that is why only big pharmaceutical companies are traditionally involved in drug discovery programs. This is a quote from Anthony Fauci. So I think you must have seen his face during the pandemic very often. He was the one of the main persons involved in the task force for COVID control in US. So he made a quote: when a company is fairly certain of a profit margin that is substantial, it can assume responsibility for the clinical trials to develop a blockbuster drug. So what he's essentially trying to say is, Usually pharmaceutical companies will only go and target those those compounds which they know that the investment that they put for bringing out all those trials, it's very substantial, but then they, as much as they invest, they are able to get the return on investment. Only such compounds are taken. This is the sad reality. That is why there are certain, for certain diseases we have a lot of drugs, because there is a huge market for that. But for many diseases, which are also called as neglected diseases, okay, we do not have much of research done because the market for that is very less. Or these diseases are more common in third world countries. It's more common in Latin America. It's more common in East Asia. So these diseases, especially tropical diseases, which are not affecting Western Europe and US, so then there is no market for these drugs in those countries, and so the big pharma is not involved in this research of these compounds. But for common diseases that is affecting the American public, like say diabetes or hypertension or coronary artery disease, these are the diseases, chronic disease, where you have huge numbers in the US, and then naturally you will have a lot of research happening on that, and a lot of products are ultimately in the market. So who are the different regulators? So every country has a regulatory authority who is involved in bringing out, giving the final approvals for the product that is being tested. So at every stage, it is important that the sponsor works with the regulatory authority. In India, we have the organization called Central Drug Standard Control Organization. So every trial that is done in India, definitely you have to get a approval from the CDSCO. Now, if it is a Western company, let's say some company that is based in US, which is bringing out a product that is going to be launched in the US, but then they are also planning to launch the molecule in India, then usually what they will do is they will conduct multiple trials which happen. They'll conduct multicentric trials that will be happening at all the countries which they are planning to launch. Particularly, the Indian regulator usually insists that for every molecule that is going to be approved, that is going to be marketed in India, the company should also include patients in the clinical trials. Only then they will give approval in the market. For example, a company plans to bring out a novel anti-diabetic drug. They have to include Indian patients in the phase 3 clinical trial if they want to bring out the molecule in India. The only exception to this is molecules which are, for example, life-threatening diseases where there is a real urgent need. In that case, the Indian regulatory authority might allow the compound to get marketing approval without performing a clinical trial in India, on the exception clause that this is a drug for an emergency, so we don't want to waste time and do one more trial in India. But on the basis of trials from Western countries, we take the data and market the drug. And of course, safety reporting has to be done. So if a trial is done in India, irrespective of who the sponsor is, we have to get the CDSCO approval. But at the same time, please remember that should we follow the FDA guidelines? Should we bothered about EMA guidelines? Yes. If the data that we are generating for a clinical trial, if that data is going to be used for getting marketing approval of a drug in the US or Europe, then our data is liable for inspection by the respective regulators. Very often you find FDA inspectors coming to Indian hospitals to check the validity of the data that is generated from a particular clinical trial. And sometimes they might come and say, no, we are not happy with the standards, and so we are delisting this particular sponsor because they have not done the trial adequately. There are a lot of issues in the particular trial. So what the point I'm trying to say is, it is not enough if we just follow the standards that is expected from CDSCO. If the particular trial that we are involved, if it is for FDA submission or EMA submission, then we also have to follow the guidelines as stipulated by those respective agencies. And then we have the local ethics committee. Uh, Ever since 2019, when the new drugs and clinical trials rule came into existence, the local ethics committee of every hospital has been empowered very much to review different aspects of clinical trials. Because you just think in India there are almost 1,000 to, I mean, I'm not sure what is the exact number, but thousands and thousands of hospitals are there which are involved in clinical research. So if all these have to be supervised by one single regulatory agency in India and New Delhi, it is not possible. They will obviously be overloaded. So they want the local ethics committee to take care of all the ethical aspects, reviewing all the ethical issues. In fact, it is not just about ethics committee giving an approval letter. Ethics committee should also do a follow-up to see whether the protocol is being followed as it was stated. So phase one. So now I'm going to talk briefly about the phases of trial to bring out the drug discovery and development. So in phase one trials that are done after the extensive preclinical animal testing and in vitro testing, we tend to begin the phase one trials, which are usually done in a very specialized centers uh, to bring out the safety of the molecule. In fact, the main objective of doing a phase one study is to find what is the maximum tolerated dose uh, beyond which we should not try to venture in the subsequent phases. So pharmacokinetics is also identified to study the absorption, distribution, metabolism, excretion. To study all these things, we do this phase one clinical trial. So phase one, we do not test for efficacy, but we test for safety and pharmacokinetics. And if you're dealing with a new molecule, this new molecules, since we are not sure, sometimes a drug might be extremely safe in animals, and then you might not find it safe in humans. So then it becomes a problem. So we go for something called as sentinel dosing, where you just give one patient a dose and then see how he reacts. And if it is fine, then we go for the subsequent full cohort of patients, volunteers, where 3 people are given the dose and see how they react. If there is no adverse events, then another 3 people are given the drug, and in this way they just keep increasing until 1/3 of subjects develop toxicity, and that is called as dose limiting toxicity. At that point, they will stop the further escalation of the dose. So then we have single ascending dose studies, multiple ascending dose studies, where you see multiple doses, what happens to the pharmacokinetics and safety. And we also look at does food have any interaction with the drug? Does the pharmacokinetics change with food intake and without food intake in fasting state? Because we need to know whether food will interfere with the drug's PK. And please remember that this phase one trials can be done only if you have completed all the preclinical safety data, like acute toxicity, chronic toxicity studies. Then usually chronic toxicity, because it will take a longer time, they might not insist in phase one. Then male reproductive toxicity, female reproductive toxicity, genotoxicity, carcinogenicity, all these studies are required. In India, one peculiar thing that is happening is male fertility testing is usually not done prior to phase one trials in most of the countries. Fertility tests and all, they take more time. So by the time you have a basic idea of the acute toxicity and subacute toxicity, and if the data is fair, they usually proceed to phase one trials. But in India, the regulator insists that male fertility testing data should be available. So because of this one reason, you will not find many countries approaching India for bringing out phase one studies. So that is why phase one studies are very less in India compared to what is happening in Western Europe and America. You find India mainly as a participant in phase 3 clinical trials. And of course, they can be done in healthy volunteers. Most of the times it's done in healthy volunteers. But in drugs which are highly toxic, like anti-cancer drugs, we usually do it in patients. The dosing for conducting a phase 1 studies is on the basis of the animal studies, no observed adverse effect level, or on the basis of minimum anticipated biological effect level. On this basis, they try to do phase 1 studies, the starting dose. And then we go on to phase 2 to identify whether the proof of concept, because this is the first time you are doing the study in patients, you have to be sure whether really the drug is working or not. So the proof of concept that the drug is working, you get to know in a phase 2 trial. Phase 2 trials are usually done for a shorter period, and many times we use surrogate endpoints where we study the effect of the drug on a particular biomarker to evaluate its efficacy. Promising results seen in phase 2 will only make the company believe that they can this drug is worth it to continue to the next phase. And phase 2 studies are usually done in a few centers. Very often it is done in the origin country of the sponsor. Like if it is a US based company, they would prefer to do it within the US state itself. And it is done in a very homogeneous population. That is why you find phase 2 trial, the exclusion criteria, you will find almost 25 or 20 exclusion criteria, because they want to ensure that a highly homogeneous population is given the drug. Okay. Now, at the end of a phase 2 trial, the sponsor comes to a very critical phase where he has to make a decision. What is that decision? It is a decision which is called as go no go decision. What is this go no go decision? That is, the sponsor has to take a call whether the data that has come so far from phase 2 studies, is it promising enough for him? Can he predict what will happen in phase 3? So unless you have substantial efficacy in phase 2 trials, many times, even though there is some benefit that is seen in phase 2, they might not proceed to phase 3, especially when you already have other drugs. In other words, what I'm trying to tell you is the sponsor will try to predict whether this new drug that is coming, does it have the potential to really beat the other molecules which are existing in the market. So this is a prediction that he has to do with his experts on the board. Now we come to the confirmatory trials, which are phase 3 trials, which are done to establish that the drug is really working. They are also called as pivotal trials because they are the most important trials to know the value of a particular drug. They're usually randomized, double-blind, placebo-controlled trials, or it could be even standard drug, which is a control. Even devices also, you could do phase 3 clinical trials. They are called as pivotal trials. And then we have usually phase 3 clinical trials. Very often the patients is not deprived of their standard medical treatment. So you will not find that a patient who is getting placebo will be getting only placebo. Usually if they are getting placebo along with standard treatment, one group. In the other group, standard treatment along with the new drug. So that is the conventional add-on placebo add-on study design, you call that. And phase 3, since they are very important to understand the nature of a drug, the outcomes that are chosen are often clinical outcomes. They are hard endpoints. So for example, let me give you an illustration. If you're talking about a drug that is going to be useful for hypertension, you would want to, or let's say an anti-cancer drug, it's not enough to show that some particular marker got reduced. You have to show that by giving the drug, the patient's lifespan improved. Instead of dying from 6 months, he lived for extra 3 more months. So survival benefit. You have to show improvement in terms of clinical benefit. So in phase 3 clinical trials, just showing improvement, tumor size reduced by 2 centimeter, 1 centimeter, that is not enough. You have to try to show clinical benefit. So I will go into a little bit more detail about phase 3 clinical trials. So usually phase 3 clinical trials, you start by preparing the clinical trial protocol, you select the site. Now the sponsor usually will be selecting multiple sites. It's because multinational, multicentric. I already told you why they do multinational, because they plan to launch the molecule in many countries. So that is why they target all the countries where they are going to launch the molecule. Besides that, they will also look at doing the study in across several centers. Like in India, if a sponsor is coming to India to do a trial, he will choose 10 centers or 20 centers based on the sample size in India. And how does he choose the center? Centers are chosen on the basis of many factors. But if you ask me what is the most important factor, the most important factor is basically the availability of the patients. If a center has good number of patients, although all other factors are issues, they will still try to include that site, because it is very very difficult to get patients to participate in clinical trials for various reasons, because of which uh, so choosing that is the number one criteria, whether the site has adequate patients. Besides that, the investigator's experience is also very important. A site which is already conducting a lot of clinical trials in a particular area, that site is more likely to be chosen than a site which is not very often involved in clinical trials. So the investigator's experience counts a lot. Besides that, the does the site have a team? Like for example, to do clinical trials, you need a clinical research coordinator, you need some research staff to assist the investigator. And if the investigator has a sub investigator, co-investigator, then all these things show that there is a team. It's not just one person, the principal investigator who's doing the study. And so the sponsor is more confident that that trial will succeed at that particular site. And besides that, there are some basic facilities that are needed to conduct clinical trials. For example, there should be some dedicated space allotted for performing clinical trial activity. There should be office space. There should be a room with where access to high speed internet, because all the data that is generated has to be transferred into electronic documents and sent to the sponsor. So for all this routine communication, you will need a computer with high speed internet, scanner, copier, fax. Routine things that are used for clinical trials are important. And if you're processing samples, you would also need a lab. That lab should have NABL or any other standard accreditation lab should be there. And the ethics committee that is there in the site should be registered under CDSU. So these are some of the things that the sponsor sees to choose a site. Phase 3 clinical trials are very often have large sample size, because they are based on the primary endpoint, and many times primary endpoints, I told you, are clinical endpoints. Like when we were doing a particular clinical trial some years back, it was a trial to study the efficacy of a drug, whether it can prevent recurrent heart attacks in a patient who has already had a single heart attack. So for this, they had to give the drug for a period of 5 years. So you can imagine that giving a drug for 5 years, so and to see how many of them go on to develop death or recurrent myocardial infarction. So the total sample size for the study was 18,000. It was such a huge number. And very often, and even in the COVID studies, you would have noticed for phase 3 Covaxin trial, the total sample size was 26,000. For the Pfizer trial, it was 45,000. So huge sample sizes are needed. And that is why the investment is also very, very huge. And that is why multinational pharmas are very often involved in this rather than smaller companies. And phase 3 studies are usually the drug is given for a longer period of time. It's not that you give the drug for just 2 months and then see some improvement and then see, okay, stop. Because chronic diseases like diabetes, hypertension, coronary artery disease, cancer, these are diseases where patients have to take the drug for a long period of time, probably years together. Okay. In fact, the standard practice for cardiology trials is 5 years, because 5-year outcomes need to be known to see the effect of a drug, whether it can reduce the 5-year mortality compared to the standard drugs. So it's very common to find certain trials having long duration, okay, in phase 3 rather than short duration trials which are seen in phase 2. Feasibility studies are done that to identify which are the ideal sites by sponsors, and then regulatory approval is obviously needed before you start this phase 3 trial. So the regulator will approve the protocol, and the regulator has to also approve each center. Like it has to say, okay, this particular hospital has the correct investigator who's qualified by training, experience, as well as the site is also capable. So they should also give approval of that particular site, as well as the local ethics committee of the individual hospitals. So like if 10 hospitals are there, you need to get approval from each of the ethics committee. You cannot say, I will just get a central ethics committee approval. It has to be approved at each committee. And the population that is chosen in a phase 3 trial, it is also having very strict inclusion exclusion, but then compared to phase 2, the population is a little bit more heterogeneous, and sample size is mainly driven by the primary endpoint of the study, because you will have multiple endpoints in a phase 3 clinical trial because you're spending millions of dollars. So naturally the company will want to try to study as much as possible from one single trial, and so they will have multiple secondary endpoints, they will have multiple exploratory endpoints also, but the sample size is purely on the basis of the primary endpoint, which is usually a hard clinical endpoint. Please remember that the choice of endpoint is very crucial, because if you choose certain endpoints, the sample size accordingly will change. Okay, so you should choose the correct endpoint. Secondly, you should choose an endpoint that you expect to see a substantive difference with the drug. So not all drugs will have effect on all endpoints. Some endpoints, certain drugs might have benefit, some endpoints the patients might not have the benefit. So you have to choose which is the ideal endpoint to be studied as the primary endpoint. And for most of the common diseases now, there is almost something like a norm followed as to which should be the primary endpoint, and so you will also have to keep that in mind. And please remember that large clinical trials which are done for several months, several years together, will frequently have interim analysis done at periodic intervals. This is for many purposes: to see how safe the molecule is, to see how effective the molecule is on the basis of interim results. And there is something called as a DSMB. DSMB is Data and Safety Monitoring Board. So this Data and Safety Monitoring Board is an independent board which reviews the evidence that is generated from the interim analysis, and they will give recommendation whether it is safe to continue the trial or this trial should be stopped. Okay, and for individual patients, there is something called stopping rule. For example, a patient is part of a trial, and then you find that in spite of taking these medications, his lipids are uncontrollably high, and so they may decide to stop the patient from the trial and switch on to standard or regular treatment. So these kind of stopping rules are always kept, and there should be a procedure to handle dropouts, how to handle dropouts when you're designing your protocol. So may I know how much time I have?
Ma'am, sir, the time is almost up, sir.
Okay, then I will try to be fast here. So the phase 3 trials are
You have some 10 minutes more, sir.
10 minutes.
15
Okay,
minutes.
fine. I'll finish it in 10 minutes. Thank you. Phase 3 trials, it starts with usually once all these approvals are done, they have something called an investigator meeting, wherein all the investigators are called together and where they plan and they are trained as to how the particular protocol should be executed at the site. The products which are going to be studied are shipped to the different hospitals, and it's very important that the product is stored in a place which has restricted access, which has temperature control, and how to handle the product, this has to be taught to the investigator and investigator's team. So the site initiation visit, which is basically the visit wherein the sponsor essentially activates or ignites the respective hospital sites to start recruiting patients. So once you start study recruitment, where you start screening of individuals, you start screening of individuals. So screening involves basically you might have to do some, you have consent, and then do certain investigations to find if the patient fits the criteria. And if he fits the criteria, you allow him to go through a run-in period where he takes the medication for a period of 10 days or 14 days, see how much he's complying with your instructions, and then at the end of that run-in period, you ask him to come back to the hospital, and then you randomize him. To receive either the new drug or the control drug. And this randomization is these days done with something called as a interactive web response system. So basically, it's a, you have to go to a website, enter the details of the patient, and then the you will get the allotment as to which sample box number you have to give to the patient. Or there is a traditional envelope method also followed in some cases. And the data that is collected all should be entered in something called as a case report form. These days, most of the sponsors, they are going for electronic case report form. And each clinical trial usually will have a fixed number of visits, let's say 5 visits or 10 visits or 20 visits. So you should train the patients to come to the hospital for each study visit, remind them in advance so that they don't miss out any visit. In each visit, there will be some standard procedures to be followed, like some standard investigations may be there, some specific examinations may be there. So all those protocols should be followed. Any change in these protocols is considered as a protocol deviation. Remember, when you do a regulatory trial, any protocol deviation is considered very serious, especially if that protocol deviation affects the safety of the patient. So then you have to take appropriate corrective and preventive action, and also inform to the ethics committee as well as the sponsor. Remember, when a clinical trial is done, you will be subject to monitoring by the sponsor, and sometimes even audits will be done periodically. And ethics committee also has the power to come and audit the site. You cannot say the ethics committee, no, you already approved the study, why are you coming and checking? So they can come anytime and check the files. And remember, safety can also, there can be safety issues in clinical trials. After starting the trials, some patients might have some side effects. So it is a responsibility of the investigator to manage the case. You have to see that the patient's safety issues are taken care of. And if it is a serious adverse event, what is a serious adverse event? Any event that can cause death or hospitalization or permanent disability or congenital anomaly, all these things are considered serious adverse event. If these things happen, the patient may be liable for compensation, and there is a guideline as per the new drugs and clinical trials rules how they should be compensated. So data clarification, resolution of the queries that are there in the data that is sent to the sponsor, that has to be done by the site. So after all these procedures are done, finally the database is locked and the data is analyzed, and the results are presented in major conferences and journals. Data is also submitted to the regulatory authority, and final approval is given with specific labeling that the drug is approved for a specific indication. So then we come to the phase 4 studies. Phase 4 studies are basically done after the drug is approved in the market. They are called post-marketing surveillance studies. Usually they're observational in nature. They are useful to collect safety data because it is being done in a large group of people in phase 4 compared to the phase 3 and phase 2 trials. You might get new safety information which you are not aware so far. So that might even require labeling change. There are many drugs which were thought to be safe when they were approved, but then after getting released in the market, they had to change the labeling because they found that this drug in a particular group of patients, it increases the risk of suicide. And so any patient who has psychiatric disorders, it is better that they do not take this drug because it can increase the risk of suicide. Okay, so these kind of modified labeling will be needed. on the basis of this post-marketing safety data and long-term safety data. Maybe the clinical trial was done only for 2 years, but then when you give this drug for a period of 3 or 4 years, the certain people have chronic kidney disease. So these kind of new data can always come up in the post-marketing period. So data on special populations can be generated which were not studied in earlier trials, and rarely you can also find extension of indication. For example, there is this a drug which is a diabetic drug which is called as Sglt2 inhibitors. Now this Sglt2 inhibitors originally developed for diabetes. Today it is being developed for heart failure and heart disease, thanks to the fact that people have identified that in diabetic patients who had heart disease, they were trying to they were seeing exceptional response. So you can understand new things which you did not know at the time of phase 3 phase 2 trials, and thereby increase the indication capacity for that particular drug. And please note that the company has an exclusive license to manufacture this product until the patency period is completed. But once the patency is over, any other company can make a copy of that, which is called as a generic molecule, and they can also market it. But to do that, they have to do something called as bioavailability, bioequivalence studies, and prove that their formulation, generic formulation, is bioequivalent to the reference product that was the original product discovered. And if they show that the pharmacokinetics, the bioequivalence is fine, it's equivalent to the parent molecule, then they will get marketing approval for that molecule. So to summarize, I would just say clinical trials continue to be the gold standard of evidence-based medicine. Most of the guidelines that we see is based on well-designed, robust phase 3 trials with hard endpoints, and good clinical practice that is followed while doing clinical trials. Only following GCP promotes scientific integrity, makes people believe that data, and of course, you're having ethical standards so that the safety, the right safety and well-being of the patients are taken care. Ethics committee has a key role to play in this process so that GCP is followed not only in letter but also in spirit. And NDCT 2019, it's a huge leap from Schedule Y which we had in the previous decade. Current guidelines, it's the current guideline for clinical trials with new drugs. So I tried to do justice to this topic, but if there is anything that you still have questions, please feel free to ask now.
I have to say, sir, you never disappoint us. Thank you, sir, so much for clearly walking us through the basics of clinical trials, the different stakeholders involved, and the different phases. And you have very briefly, but you have tried to bring across the major challenges you have faced. So I am looking at the chat box. I'm not seeing any questions. But sir, I would like to ask one. So what was one of the biggest challenges you faced when you got into this scenario of going in for clinical trials, of becoming a center, and now you're a consultant and you give a lot of advices? What was the first thing which, you had to cross over to get into this?
Okay, so yeah, thank you. So clinical trials, even though we know as doctors, we know that clinical trials are important. Without clinical trials, you cannot bring out new molecules. The sad truth is that in India as a whole, research is not looked at in a very positive way by patients. The people, in fact, today, and the sad thing is that it's even changing for the worse with each year in passing. Like in 2012, if you were to talk to patients, they view the way they look at hospitals, the way they look at doctors is not the same as how they look today in 2022. You know that very well. So things have changed. There is a trust deficit that is happening in healthcare as a whole, and that trust deficit is affecting clinical trials also in a large way. Even though we tell things, we explain things to patients, not everyone have a very positive attitude towards research. So it is not easy to recruit patients. It it's very difficult to follow timelines. The sponsor will say a timeline, look, we need to recruit this many patients within this time. But then there are patients who are not fitting the criteria. When before the trial begins, we feel that we can complete the trial as per the protocol. These kind of patients visit our hospital. But then when you start actually doing the study, you realize, number one, many patients invariably get excluded because of at least one or two exclusion criteria. And the other aspect is the patient's willingness. Sometimes patient is fitting all criteria, but then this one thing is there, not willing to be part of study. And sad thing is today, this unwillingness is seen even for observational studies. Forget about clinical trials. Even studies where you're just taking 2 ml blood sample to do some biomarker estimation, even for that there is unwillingness. They don't trust that the 2 ml of blood sample they give is going to be misused. So that is the state we are living in now. We are becoming more and more like how it is in the West. So altruistic mindset is not something that is commonly seen. There is a need for public engagement in these issues. Clinical trials is a bad word, basically, in many places. It's a taboo to be part of a clinical trials. People they see that clinical trials should be done only by people who are totally impoverished, who are totally dependent. Only such people to be part of clinical trials. That should not be how it is. See, today we have so many drugs in the market. How do we have all these drugs? It is because somebody in the US, somebody in Europe was a clinical trial participant. They understood the risk and they took that risk. When I was doing phase 3 clinical trials with Covaxin, I used to go and conduct camps to make people aware of this Covaxin trial, be a participant. And one of the questions that people asked, have you taken the Covaxin injection? So then me and my colleague, we discussed and we felt, okay, the only way these people are going to believe us is if we ourselves be participants. So we wrote to the sponsor and the sponsor said, okay, because you're co-investigators, you can definitely take in. And we enrolled ourselves in the trial, even though we were investigators in that trial. And then we came back and told, look, I have taken it, so now you take. So we had to go to that extent. So that trust deficit, I'm sorry to say that we need more public engagement. People should understand what clinical trials are and be happy to participate in it.
Thank you, sir. Thank you so much. So there's a difficult road waiting for people who want to take up research in human beings, and even now animals also. It's going to be very difficult. So I don't know how we can bridge the gap of that translational research. So thank you all.
Thank you. Welcome.
Thank you so much, sir.
Thank you, Dr. Shirley, for moderating this session. And thank you, Dr. Melvin, sir, for such a insightful session. Thank you, sir. Now we move on to the next session. I invite once again Nebu sir to moderate the next session and invite the speaker.
Thank you, Dr. Shirley. So it is indeed a great pleasure to welcome Rajiv sir. Sir was our former professor in community, head and former professor in community dentistry or community medicine of Pushperi Institute of Medical Science and Research. And sir was one of the initial pillar of research in Pushpagiri. So it is indeed a great pleasure to welcome sir to our second session. And sir is currently the head of the Department of Community and Family Medicine in All India Institute of Medical Science in Andhra Pradesh. So sir will be taking a session on the translate the finding from the practice to the population. Sir, welcome to the session, sir.
Thank you. Dr. Melvin is still there?
Yes, sir. I'm there.
Yeah, I have two questions from your presentation, just
Yes,
for continuity. I typed it in the chat box. If maximum tolerated dose is less in Indian population, when we do the phase 3 first time in India, and then we find that the dose which was found out as maximum tolerated somewhere else was actually not tolerable in India. Do we have to do a phase 1 trial again to complete the cycle again, or we reject the molecule altogether?
Okay, great question, sir. So this is something that is very often seen in clinical practice, that the dose that is written in standard textbooks, Western textbooks, is not the dose that our patients tolerate. Very often this is seen with beta blockers and many other drugs. So as such, in clinical practice, there is this thing to bring down the dose and all that. But these days, when it comes to clinical trials, if a molecule, let's say a molecule has come to India for phase 3 trials, already phase 1, phase 2 is completed in Western soil, and then you find there is a lot of tolerability issues, that will be in the first interim analysis itself, that will be picked up.
Yes.
And if it is picked up that patients in India are really not tolerating this dose, they would already be having the earlier dosages experience in the Western data. Based on that, they might uh, either modify the protocol, come back to the drawing board and say that this is the reason we want to give a much lower dose, and so we want to modify the protocol for the Indian subpopulation of the study. So that is possible, but it is not a very common thing, because most of the time, by the time the phase 3 trial comes, at least this much understanding about the drug these days, they are able to know how much the people are able to tolerate. But then it is always possible, it can be done, the protocol modification can be done.
Thank you.
Yeah.
Thank you. The second question was like, in the phase 4, if you find that there is a new indication, repurposing, is it phase 4 study or phase 3?
Yeah. So if a new drug, I mean, if a drug that is already in the market now, we are trying to identify it for a new indication. If the drug company is going to uh, submit the data for marketing, he has to do it as a phase 3 only. Like for example, the example I gave of Sglt2 inhibitors like empagliflozin, dapagliflozin, they were diabetic molecules, but then they came back and started doing clinical trials in heart failure, which are phase 3 clinical trials. So they have to get regulatory permission for doing phase 3 trials. So it will be phase 3. But suppose if I, as an investigator, if I am doing a study with dapagliflozin in the market in a small group of patients with heart failure, 50 patients, that will not be considered a phase 3 trial or a phase 4 trial. That will be an academic study, because I will take the data and I will publish in some journal, and I will say, look, in the 50 heart failure patients whom we gave this drug, this was the response. That's it. I cannot use that and ask regulator for marketing or anything of that kind.
Okay, thank you. With that, I think I'll go on to my presentation. Thank you, Dr. Melvin, and thank you, Dr. Nebu, for the fine introduction. And Dr. Soumya, I think this there on the host part of it. I think I am allowed to share the presentation, right?
Yes, yes, sir.
Okay. Okay, let me see. Okay. Okay, I'm the co-host. Okay. Okay, that brings us to the. Sorry. Technical. Okay. Okay, that brings us to the two things which was spoken by the previous two eminent speakers, starting with Sabu Thomas sir, who said there is a fourth part of to every translation, like from going from bench to bedside, and from bedside it has to move into the population. I don't have a smart word for the B. If I may, can I ask the audience to say what was that particular term which Sabu Thomas sir had pointed out as some 1, 2, 3, 4? Can you all type in the chat box? Just to see, if how many people are alive. Come on, guys. Okay, I think I'll move on. How many of you can actually name the person who is actually indicated in that picture over there? If the picture is visible, it's a little dark. It's like a pencil drawing. How many of you can actually name that gentleman who started a revolution, which we are seeing the benefits in the COVID era itself? Whatever Dr. Melvin said about the community not being part of or being very keen or being large-hearted to be subjects of clinical trials or even simple as vaccine trial. Who is that particular person who is on the screen, who is actually seen giving some kind of needling to people who are there are onlookers, there are people who are waiting, and then you see that particular person brought about a big change in the field of public health, if I may call the whole thing as public health. Okay, he was indeed monitoring the health state of the people, and he was finding out that there are some illnesses, especially one illness which was causing a lot of disfigurement and probably deaths in the community, including stigmatization, patients getting thrown out to the villages to die a rotting death outside the villages. All that was noticed by that gentleman. He found out a community health problem in which he had something to do. He investigated it, and he found out the health hazard, and then he found out that some people are immune to this particular condition, and the diagnosis, which is nothing other than smallpox. And the gentleman is nobody other than Edward Jenner. And he found out that the immunity provided by cowpox can actually be translated to protection against smallpox by simple observation. Actually, not his observation, somebody else observed it, and he found out that he can actually courageously go ahead and do something about it. He educated, and empowered the people that the word vaccinia comes from his experiments. The vaccinia, the word vaka, meaning cow. So whatever cow gave to the milkmaids, he extracted the serum. Or nowadays the opponents of vaccine, this thing they call it, you're taking pus and making it into a vaccine. How allergic that can be, but then what he did was he took the serum from the smallpox cowpox lesions, and he he did the vaccinia scarification, so to speak. It was not even an injection. You put a drop of the serum onto the skin of the people, and just with a multi-pointed needle, he'll just scarify the skin, so that the injection is almost intradermal, like we are giving BCG and rabies vaccine nowadays. So he succeeded in preventing smallpox from population, and he mobilized community partnerships, and he found out action, and he tried to solve the problem. And he almost managed to make it into a policy which made compulsory vaccination a kind of a regulatory requirement for the population. So much so, we are moving to the next part. Whatever Dr. Melvin was telling, that opposition came. Okay, that's where we have to move from public health to over a century later, probably, to population health. And you can see the networks which are active in population, starting with the public health agency, which is trying to make vaccine compulsory and trying to roll it out. And you have people who are in the drug treatment, law enforcement, doctors, tribal health, faith institutes, who might be favoring or opposing the vaccination strategies, non-profit organizations, nursing homes, community centers, fire, transit. You saw every kind of community action group coming into force during COVID, and civic groups. Mostly these are the people who might be actually creating a bit of issue, when it comes to making it compulsory. So when the law enforcement, the government actually tries to make it into a law or regulation to tell that everybody should be taking smallpox, without smallpox, or without COVID vaccine, and nobody should attend a school or something like that, then the opposition starts from all these other partners in the whole game. And that means that we have to move from probably public health. In fact, public health was, made famous by one gentleman known as Chadwick, who was a hard taskmaster, hated by many people. And that actually brings us to the, requirement that, we have to link people. We have to link people to the needed personal health services. We have to make sure that, they require it as something which is their right and not forced on them. And we have to provide the healthcare when other people. Otherwise, it will be unavailable. You have to make it free. You have to make it, offers. You have to make it open, and then you have to assure that a competent public and personal healthcare workforce is actually involved in the thing. We have seen everything in the last 2 years. All those things being rolled out in the face of the calamity that is the pandemic of COVID-19. And then, of course, side by side, we have to find out the effectiveness, accessibility, and quality of personal and population-based health services. And also, so much of research went into a disease in such a short span of time, so that all this NDCT 19, the Schedule Ys, and the other kind of processes which used to cause efficient, eminent kind of delays in the marketing, rolling out of products and all that, just evaporated. It became an emergency. It became emergency use authorization. Many of the technologies which were hiding in the dark for fear of people's opposition, they came out into the public front, and we see that many of the 20th century achievements which we have seen actually got translated in a short span of time, so that we are actually breathing free now. We are able to move around, even though this is an online sort of session, we could have done it even without, being away so many kilometers away from the main site. Okay, so we have seen that immunizations have gone through a lot of cycles. Edward Jenner was hated, actually. People threw dead rats into his compounds. They fought him like anything. And then when motor vehicles came in, there was no safety at all. Initially, there was no safety because it was a necessity. People thought, dying is a part of motoring around. So later on, we found out that people came out with, protective devices, warning devices and all that. And of course, we know that workplace safety became a high priority. Otherwise, people used to die in mines and all that, and control of infectious diseases. For some time, we thought that we have conquered infectious diseases until COVID made a comeback. And then we found out that deaths from heart disease and stroke is eminently preventable. We found safe and healthier foods. We are trying to avoid the bad foods nowadays. We are finding that mothers and babies are healthier. Family planning is succeeding. And of course, in the field of oral health, fluoridation was a major game changer for preventing caries and all that. But then, like the life cycle goes, each discovery later on will be found out that there must be some problem associated with, and then we found out fluorosis as another natural issue, wherein probably fluoride toothpaste became a villain as well. And tobacco, definitely it's a health hazard. So where are we now? From the last century, we are moving on to the next century. We are finding that healthy people requires access to health services, because the gain in prevention of heart diseases, stroke, all that actually has not been transferred to the population. It has come to the bedside, but it has not gone to the population as yet. So much so, some of the sections of the community are still suffering from a high mortality rate. Even in COVID, we found out there was a huge gap between the death of upper class people and the lower class people, even though it was reduced by active intervention of so much of public health agencies and all. We found public health as a successful endeavor during COVID, and clinical preventive services, has to be rolled out, and quality has to be assured. You have to be protected from injury and violence, which actually found a little bit of increase during the lockdown period and all that, and MCH became a priority. Infant deaths have come down, according to the latest SRS reports. Mental health is becoming a big issue, that too. With the COVID, we found out lockdown anxiety, lockdown psychosis, all those things came up. Nutrition, physical activity, and obesity are frontline issues nowadays. And of course, in the healthy people for this millennium, we are finding that oral health also takes a front seat in the top 10. Reproductive and sexual health and social determinants of diseases have come to the forefront. That might be the slow shift what we are talking about from public health to population health. Social determinants. Kerala has been talking about social determinants for the last 5 or 6 years. Okay, so we are a little bit ahead of other states when it comes to rolling out population health compared to probably other states and all that. Substance abuse, tobacco still retains one of the top positions. So what are the community health initiatives which you can talk about in population field as the backdrop of this particular online session is all about medical dental integration to those people who can afford it and those who cannot afford it also. We have to integrate oral health services into the primary care. We are struggling to get doctors into the primary health center. We know that. We are yet to see dental services becoming a routine activity in a primary health center. We have to think about it. The health systems have to improve. And of course, for the people who are eating too much, probably dental issues are much more, non-communicable diseases as occurring as secondary to the dental conditions. We know about it, periodontitis and other conditions. Okay, but then when people are poor, probably we have to provide community food hubs which are providing healthy food to people also. This also is another aspect of socioeconomic determinants. I'll be talking about it in the next slide. High quality, healthier foods for hunger relief to people who cannot afford it, and of course people who can afford it, but they can't take it on their own. People who are old, the proportion of old people is increasing world over, and especially in India, especially in Kerala, people living alone in old age, especially single old people living alone. Probably we have to think about from the societal point of view, family caregiver system, so that they are not feeling alone. They are being able to eat, get the food, and probably they need help to eat also. That means that some amount of insurance should come in, either the government way like the Ayushman Bharat initiative, what you're talking about, so people have to be protected against, all this kind of negatives through interventions of the community, so that will bring the population health into focus, and that actually see some of the successes which I want to highlight over here is like vaccines for children. When I was a student, and when a current MBBS student is concerned, we see that some of the illnesses which we prevented using vaccines, the new students don't have any exposure at all. So it has been a huge success. We are trying to introduce more vaccines, into the national immunization schedule, and that's a success story in spite of the opposition which Dr. Melvin was talking about in terms of translating the bench findings to the bedside. And then lead poisoning, which used to prevent our young children from attaining the cognitive development. Now we are very happy about the world chess champion from Tamil Nadu. Okay, so without this prevention of lead poisoning, probably we may not have seen Pragyananda achieving such heights and all that. Lead is actually a very small micro kind of poison which can actually destroy your brain function, so that a couple of points of your IQ can be deleted from the overall population. And then tobacco, it's a big menace, but then COVID changed everything. So COVID-19, was it a good thing or a bad thing? And some of the authors say that they can describe the COVID-19 as best of times or the worst of times, whichever way you want to take it. So either the cup is half full or the cup is half empty. How so? So before jumping into that, the backdrop which I provided so far, and with the discussion with the previous speakers, we see that population health is the fourth domain of translating any research findings in the lab to the population side. It's the health of the population as measured by health status indicators, as influenced by social, economic, and physical environments. And then that also emphasizes on personal health practices. We don't ignore people. We provide individual capacity and coping skills, and we provide human biology background to the whole discussion. And of course, we also have to talk about primordial prevention in the form of early childhood development. If you want to go into the womb, okay, you can even talk about, in fact, some of the researchers pointed out that probably what the mother is eating during the uterine life of a baby actually orients the child to a particular flavors of food and all that, so that means that we have to go even before childhood or infancy into the womb, so that you can change the dietary habits of the children or infants or babies, okay, so that you can even affect the level of oral health of a community. And of course, provision of health services is part of population health. So this is a definition which I can give you. Health of a population as measured by the health status indicators, influenced by socioeconomic indicators, physical environments, personal health practices, on to early childhood development and health services. Okay, COVID-19 and infodemic, the scare which was produced by COVID-19 and the aftermath of it, Dr. Melvin commented on it. The fear of a novel virus it started off with, and then it resulted in social disruption, and then suddenly total mistrust of science happened because of rapid advancement in the knowledge which was being output by frantic research world over, things changing on a daily basis. Some people said some of the medicines were good, and after a week they said it's bad, then they said azithromycin is good, then they said remdesivir is good, antibodies are good, cocktails are good, that mab is good, this mab is good, so people started seeing 20 years of development packed into 2 years. They started mistrusting science, but then in the last 20 years, they did not see what was happening. But in 2 years, it was a fast forward. It actually resulted in a bit of mistrust of science. You open Netflix, you open Amazon Prime or whatever it is, you see a lot of movies in which doctors are, or the pharmaceutical industry is painted as villains. Okay, so mistrust of science is another phenomenon which is happening. We have to remove it. But all said and done, COVID-19, the best of times, showed that, many of the technologies which was, hiding in the dark because of fear of, population backlash, was rolled out because of desperation, and it actually reduced morbidity and mortality after the impact struck. So from the social determinants, we see that population health can evolve and become a new paradigm. This approach utilizes non-traditional partnerships among different sectors of community, like public health. So public health becomes one part of it. Industry becomes another part. Academia becomes another part. Academia fought for and against vaccines. For example, healthcare industry, healthcare fought for vaccines. Local government entities were frantic to implement public health policies, come what may. Not fearing backlash and all that. They worked selfless, and we see success at the end of the tunnel, as WHO has pointed out yesterday. So we need to achieve the positive health outcomes. How do we do it? We look at the social determinants. We need economic stability, which was disrupted during the pandemic. We need safe housing, which was required for, safe quarantine, isolation. We needed access to quality food, all the time, especially for all people. Healthcare, access to quality healthcare services is a prime determinant of death rates. Even in the presence of the best of public health, you see that the top class will be having 1/4 of the death rate of a lower class. Okay, the so-called Attapadi syndrome, whatever it is. Okay, access to quality of health services, and of course education through mass media and emerging technology. Of course, formal education, quality education, and of course once you are passing out, you need quality employment. And then society has to be safe. Public safety and public transportation services are also important determinants of health. So coming back to population health, okay, the story which was told many times and referred to Merck 2018, a man and woman fishing on a riverbank sees a woman struggling in the current. This is like medical care. Patient is brought to the hospital, they rescue her in an ICU, and then they see another man struggling, they bring him in, and then finally the ICU becomes full, and the man and woman becomes desperate. They go upstream and find out what happened. And they found out, people were falling in where they were all going to a tourist spot where, there is an overhang, which is not protected. People are just falling into the river and then getting washed away. So they went to the community, like a population health paradigm. They went to the community leaders and found out, okay, the solution, long-term solution will be not saving them in a heroic fashion, but to give a protective guard and post warning signs in that dangerous place. So preventing a problem saves resources, energy, lives. But sometimes preventing the problem can become another problem, because we saw it in vaccine also. We saw it in COVID also. Even though miracle drugs and miracle vaccines were coming out of the shade into the open sphere, we found that many of the countries were not able to afford it. When America was being protected, 50% of the population, you found some countries like Ghana and all that, there was not even one person who were protected against COVID. So the population health will have to address this issue also, the issue of equality, moving to equity, providing more resources for the less privileged, underprivileged, so that they also can enjoy the nature as it is, can enjoy the world as it is. They can also move around in the same fashion. So moving on to prevention, we see that even though public health is trying to provide all these things, homeless people, you enumerate people at the homes and all that, you leave out homeless people, the beggars on the street and all that, their death rate will be still worse even if we were to do 98% of the work. Food and water insecurity will be a problem. Unemployment will be a problem. This all results in high morbidity. In spite of the technology being available for the last 100 years, we see that people are getting all this kind of problem, whether we talk about primary prevention, we talk about secondary prevention, tertiary prevention. Primary prevention, which includes eliminating the risk factors and providing health education, immunization, including probably screening, early screening. We found a lot of intervention during COVID. Secondary prevention, early diagnosis, and then treatment of the disease. And tertiary prevention, which talks about eliminating the disability associated with advanced disease. Okay. And then we have the primordial prevention, which we are talking about early childhood activities, which can actually prevent even chronic diseases in the future. You're born with a low birth weight, you can go on to get all this diabetes, hypertension, and all kinds of non-communicable diseases. So psychiatric illnesses, social illnesses, alcohol dependency, you name it, they all have an origin around the time of birth. So we have to target this also. And in oral health, this is especially very relevant. Okay, so I'll bring in a new idea of mine. Dr. John Snow, who researched, I'll show you in the next slide, who researched and made a lot of observation on cholera in England in a century where there was no knowledge of bacteria and all that. He is called the father of epidemiology, found out the connection between many things in London to the occurrence of cholera and its death and all that. He is called the father of epidemiology, but in a contrasting fashion, cholera is called the father of public health. So why don't we call COVID-19 the father of public health? In future, probably we can call COVID-19 as the father of public health. And if Dr. John Snow mapped the Broad Street area and found out that the death rate of cholera around that Broad Street pump was connected to the water inside the pump, even though he did not know about Vibrio cholerae and all that. He broke the handle of the pump and he stopped this outbreak. Like many other outbreak in London, we find that big data of COVID-19 actually helped us in mapping out a lot of COVID spots in and around the country, and then we could actually curtail it by surveillance and containment strategy, which we used in smallpox. It was brought back in COVID-19 in a less successful fashion. We were having live data coming in from all of the direction, so that you could actually drill into the data and find out something about what is going on in each area with regard to socioeconomic determinants and other kinds of things. So the public health came into fore again, and it was politicized. And in fact, public health is a little bit politicized anyway. Initially, it was a lockdown because 100,000 cases were occurring every day and all that. So we had to lock down, and then that became a big issue. And later on, suddenly all the research was fast-tracked, all that emergency use authorization as against the NDCT-19 process. We found, a lot of trials, the ivermectin coming in, azithromycin coming in, vitamin C coming in, this coming in, that coming in. Finally, we found out whatever worked in the first wave did not work very well in the second wave. And we found out that steroid is much better than anything else. Oxygen was the crying point in the second wave. And the third wave actually, in many, not in many states, at least in some other states, proved that, this result outcome actually, the translation actually worked when it came to the third wave, in which we could reduce a lot of morbidity and mortality because of the successful experimentation during the first two waves in India or elsewhere. And during all this, India approved 2 vaccines against a lot of opposition, even from so-called scientific people, the anti-vaccine group, they fought against Indian-made Covaxin. And then, of course, the Covishield came in, they fought against that also. But then, in spite of all that, it was rolled out. And most of the population in India is now protected with at least 1 dose or 2 doses, and even precaution doses, as we call it. And then now we are living in the era of variants, wherein we are doing screening, we are doing quarantines wherever necessary, we are isolating the patients, we are finding, we are licensing more and more vaccines like Corbevax, Covovax and all that, and we have found a few antivirals which looks promising, and of course the surveillance machinery is also working very hard. And all the vaccine technology which was hiding, I said before COVID-19, came out of the dark era which they were in, and we found out most of these vaccines getting some sort of acceptability in the community. We found viral vector vaccines like the AstraZeneca Oxford vaccine, which was marketed in India as Covishield. The Sputnik 5, again another viral vector viral vector vaccine came out. mRNA vaccine was successful in better, affordable countries, even though there is no point arguing between the efficacy or effectiveness of both the vaccine, they're comparable. The Pfizer BioNTech vaccine, the Moderna, which is partly mRNA, it's a mRNA vaccine again. Then we found, India had its own Covaxin, which is a whole virus vaccine, and then China had Sinovac. Protein subunit vaccine has been recently licensed in the form of Novavax. We are seeing that even the age group to which we are administering these vaccines are coming down steadily. Probably in future it might become a routine immunization campaign. Again, we have to convince the population regarding the benefit of it, rather than just forgetting and telling that COVID is over. COVID is not over. WHO yesterday also reiterated that, okay, there is a light at the end of the tunnel. That doesn't mean that, pandemic might be getting over, but pandemic going away doesn't mean that epidemics will not happen. That will happen. People who are less immune, they will die. All these things can happen. Of course, the DNA vaccine also was licensed or was given some kind of emergency use approval. The Psycho-D vaccine, the Indian product, and of course the vectored immunoprophylaxis, as is the background subject of this whole 2-day activity. We see that the new kind of technology like vectored immunoprophylaxis or immunoprophylaxis by gene transfer. Instead of you putting an antibody or antigen into the system, you just put the gene which is responsible for making the antibodies or making the antigen itself, or maybe even making monoclonal antibodies which are specific to that particular condition. And all this technology was available. Only thing is probably it was fast tracked by the pandemic because of urgent need. Millions of people dying. We have never heard about this kind of calamity in the last 100 years, in spite of all the scientific things which we are sitting on. And we see that hesitancy is still a problem. So whatever phase 1 trial you do, 2 trial you do, 3 trial you do, with all that scare which is going on, hesitancy is a population thing which we have to address. You talk about, okay, if you're talking about COVID vaccines not preventing illness, which is in fact probably a little bit true, because many other people who took vaccine also got the illness, but a milder form. It did not kill them. Okay, death was almost certainly prevented, but still that hesitancy, because hey, it did not prevent anything. No, it's not the thing. And there are people who are talking about natural immunity being better than vaccine immunity. At what cost? After killing so many people, you want to achieve it. And in that context, then why do you take BCG? Why do you take pertussis vaccine? It doesn't prevent the infection totally, but we still do take it because it has got an impact of saving a few tens of thousands of babies from tuberculosis of the brain or miliary tuberculosis, pertussis from complicating into meningoencephalitis. Typhoid vaccine is not the best one, but we still do depend upon typhoid vaccine. Cholera vaccine. Pneumococcal vaccine is, again, it should have been popular in the old age and all that, but then somehow the kind of environment what we have, people are not going for it, in the level which we are expecting it should go. Nowadays, we have doubt about rabies vaccine also, but then the veterinarians are telling that 50% of the dogs which are roaming around, the stray dogs probably might be carrying the rabies virus in their mouth and all that. Okay, in spite of that, the fingers are pointed at rabies vaccine, not at the dogs. And enthusiasm, I'm not against dogs, by the way, and enthusiasm was shown in smallpox, okay, with a lot of opposition to start with. Poliomyelitis, again, some of the experts in Kerala talked, we can't just give out polio vaccine like, giving toffees or, things like that. And then diphtheria, pertussis vaccine, tetanus vaccine has come a long way. DTP has become penta. Measles vaccine is very effective. We are talking about eradicating measles from the face of the earth. Meningitis vaccine, people do take it. So that means that, to understand the whole thing, including the hesitancy paradigm and of course the population health, you have to do a Jemba walk. What is a Jemba? Jemba means real place, the place where the real work happens, the people where People are struggling in spite of pandemic threatening them to kill also. The people who are working in the field, you go and see where the real work happens, so you can observe and analyze the process. Ask why. Be eager to listen and effort to understand the process you see. Respect the people doing the work, not talk rumor mongering against the industry and all that. Talk about the sincerity of the people who are on the field. The anecdote: Ranjidas Sagar walked each day more than 10 kilometers on foot through a forest, wading through streams to the further villages in Kurli Gram Panchayat in Rayagada district in Odisha. The journey is hard in normal times itself, but even more challenging in monsoon. She decided to reach the distant and dispersed settlements of Dongriya Kond, a particularly vulnerable tribal group, afraid of all the new technologies, the vaccination. When she reaches Dongriya Kond, she will find that the houses are empty, the tribals have vanished, they have gone into the forest, and obviously Ranjita cannot follow them. But she and her team did not give up. Finally, everything fell into place. That's the satisfaction which the Jamba people, the real Jamba work is actually going to teach you. So population health means getting involved in it, getting involved in health education about the vaccine, the technology, whatever technology, the tissue engineering you're talking about in producing the vaccine. Finally, if it has to succeed, people have to believe in it. Population has to trust in it. So population health becomes the priority in phase 4, and afterwards, vaccine has to be made available along with the trust. The people are trusting and vaccine is not given. That again creates an opposition. Population's adherence to the vaccine schedule should be watched. Many people have not taken the precaution doses probably in the high age group, or maybe some people have not taken the second doses itself. Herd immunity, you can't depend upon too much natural herd immunity because it will be at a cost. Vaccine is going to save that cost, by making people adhere to this thing. And this talks completely about disease prevention. And thankful to Rosemary M. Karen for her book on population health, epidemiology, and public health, the management skills for creating healthy communities. A fantastic book. If you get a chance, you should go through it because it's a much more detailed version of what I just presented today. So population health. Whatever pain people are talking about in terms of rolling out the medicines, in terms of, promoting vaccines, it is all very temporary kind of thing. Pain is simply an illusion. Okay, we have people at the stage of susceptibility. We have the people who are suffering from subclinical disease, which you're not detecting. That's what actually happened with Covaxin and all that. It is not, it's preventing a lot of subclinical disease, but probably it's not showing up in the symptomatic kind of diseases, so people don't believe in it. But then if you keep doing the screening, the surveillance and all that, you find that it has got 70 to 80% of the efficacy, just like pertussis vaccine, just like typhoid vaccine, just like cholera vaccine. So it's no less a vaccine compared to the other technologies. And stage of clinical diseases, which becomes obvious in above the horizon of vision of people. And of course, there are stages of recovery and stages of disability and death, which will be prevented in a large way, applying population health in a chaotic fashion during the pandemic. So thank you. And I'll stop sharing now because I'm not able to see the chat box with the PowerPoint on. Thank you. Thank you, Dr. Nabu.
Yeah, thank you, sir, for the enlightening session. And thank you for covering the aspects of community health initiatives, epidemiological studies, public health success, how it can be translated to the next levels. So thank you very much for covering almost all the aspects of public health studies. And also, sir has briefed about the COVID pandemic, how the findings has been translated to the next level. So thank you very much for sparing your valuable time.
Thank you,
sir. Thank you. So we are going to the next session. So our next speaker is Dr. Shantanu Patel. So he is the most apt person to discuss the various. He is the head of the Department of Translational Research of SRM Medical College, and sir will be talking on identify and utilize the resource available at the university level for the translation research. Thank you, sir. So welcome to the session.
Thank you. I suppose I'm audible and visible?
Yeah, audible, sir.
All right. So let me share my presentation.
Yes.
right. So number one, thank you so much for inviting me to be a part of this 2-day event. And I've been asked to speak about doing translational research in the university setting or within the university. A lot of you may be familiar with SRM University, or as it is now called SRM Institute of Science and Technology. We are a pretty large sized university. These are just some of the statistics to show you how large we are. We have almost every single discipline taught within our campus, except for veterinary sciences. We have colleges which cater to all the branches under engineering and technology, management, science and humanities, medical and health sciences, law,
Sir,
agriculture. Yeah,
sir, the PPT is not in presentation mode. Are you able to change the slides?
I am able to change the slides.
So we are not able to see that. Now it's fine. Yes, so can you put it on the presentation mode, sir? It's
I'll
not.
do it one more time. Is that better now?
No, sir, it's not in presentation mode.
On my end, it is in presentation side.
Fine, sir.
What should we do? Tell me what we do.
Sir, can you present once more? Can you?
Okay, let's do that. Let's stop and start again. Is it in presentation mode now?
No, sir. Sir, just proceed, sir. That's fine.
Okay, I'm sorry about that. I don't know. Okay, so it's so just to yeah. So as I was telling, we are a university which has all the faculties of present in this which we can be taught except for veterinary sciences. So this actually makes it a very ripe environment for carrying out multidisciplinary work as well as translational work. And this is the story of the university. I guess it is too small to be seen if we can't see it in the full screen version. So I'm going to skip this slide. It's basically a timeline of how we have progressed from a small engineering college to a full-fledged university, which is having a NAC 2 plus rating and ARIA ranking of number 2. We are also ranked extremely high in the NIRF ranking, which was ranked recently in the top 20 universities in the world, and we ranked at 20 in the medical and 8 in dental. And so this has not been something which has happened overnight. This is something which happened gradually with a very sustained and a determined effort. And the work is only half done as far as we are concerned, and we hope to be close. Scaling the ranking system as well as actually doing productive work which helps everybody out in the world, right? These are some of the collaborations which we have internationally as well as nationally, and these are some of the domains that the university had been focusing on. We have a nanotechnology research center, and we have extremely robust research work and centers of excellence in biotechnology, energy environment, and as well as in cyber physical systems. Multiple centers of excellence, multiple research centers with international as well as national collaboration. So this is something which we are quite proud of, and which we are very happy to share that has produced excellent research in the past.
Sir,
Yeah,
sorry
please.
to interrupt.
Yeah, tell me.
Sir, could you mail us the PPT? If somebody could mail it on your end, sir, we'll.
I'll do it. I'll do it right now. So you want me to stop all
Then
together?
then we can present it for you, sir.
All right.
Yeah.
Give me 5 minutes then.
Yes.
Or 2 minutes. Yeah.
Yes,
Oh, email address. Which
sir.
email
I'll
ID?
just post it.
Okay.
So please look at the chat box, sir. It's there.
Yeah, I'll do that. Okay, I've sent it, so let me know when it hits your side.
So we've received it. We're downloading.
Okay.
So can you see
Wow.
the PPT?
Yeah, I can see that.
Yes.
All right. So how are we going to do this? I say next, and you're going to do the slide.
Yes, sir. Yes.
All right. Okay. So this is like uh, repeating again, right? So anyway, we'll start from this slide. This is the slide which I was talking about. This is the story of how SRM achieved its current status. We started off as a single engineering college in 1985 and has now grown to a current status where we are number 2 in the ARIA rankings. Can you go to the next slide? We are ranked number 19 in the NIRF ranking, which we announced a couple of months back, and 20 in the medical side, 8 in the dental side, and we are a NAC A++ accredited institution. Can we move on to the next slide? These are some of the collaborations which are there with the university, and we've been working very closely with these. Some of these are universities, some of these are industries, and this has been the backbone of how innovative and research activities are happening in the university at the moment. Next slide, please. The university has multiple centers of excellence and large, and the focus of our the focus domains are listed here. Some of these are just few of the ones which we are doing pioneering and great work, and there's a lot more happening just beyond just these focus domains which I've showed you. Next slide, please. These are some of our research centers, and you can see that they span across all the disciplines of engineering as well as health sciences. These are in collaboration with not only industry but also government-aided grants have been received for setting up a lot of these centers. Next slide, please. Some of the facilities which we were highlighting in this, we have a large rapid prototyping lab for 3D printing. It has multiple 3D printers. We have a bank of 3D printers, and they're not just your FDM, the fused deposition modeling type of printers, but we also have SLA printers available. We are also working on developing metal printers in-house and also 3D bio printers. I'll talk to you about it a little later in the presentation. We have biomechanics labs, we have startup and incubation space. We are also one of the centers for the SRM DBT platform for advanced life science technology, which is providing all the facilities for genomics, proteomics, and bioimaging. We have an industry-sponsored robotics lab, and also we have a maker space, which is for what we call our fab lab, which is a center which is equipped with all the tools, machinery, and also inventory for students to come in and build their prototypes from the ideas which they may be having. Next slide, please. So where do I come into all of this, right? So I returned to India in mid-October of 2015 after about 15 years in the United States. I'm basically an orthopedic surgeon by training, and when I returned to India, I was offered a job at SRM University, where they wanted me to set up a translational medicine and research center. This is basically a story of how we got this center going and what problems we faced and how we have I'm not going to say very successfully, but have managed to set up a translational center, research center, which is now showing its outcomes and something which we are very happy about the way the things are going. Next slide, please. So you heard this term bench to bedside multiple times, I guess, over the past 2 days. And we chose about 6 different domains of research in which we thought we would be able to make a difference. The Department of Translational Medicine Research essentially partners with anybody and everybody who has something to offer which can impact the delivery of healthcare, not only for the patients, but also for the practitioners and also for the entire economic systems of the healthcare industry, so to say. We have a class 10,000 clean room facility, which basically forms the crux of our department's strengths. We do all our cell therapeutics and tissue engineering work in this cell culture lab. That was a photograph which you saw in the previous slide of our clean room facility. We are also very much interested in 3D modeling and 3D prototyping. We focus on building and indigenizing biomedical devices, and the core area of our interest is essentially related to biomechanics and computer modeling. Next slide, please. This is just a rundown of all the facilities that we have in our translational research department. It's a 5,000 square feet facility. As I told you, we have a cell manipulation lab, which is equipped with all the equipment needed to carry out cell culture and stem cell work. We have a chemical synthesis lab for all the non-sterile work, such as biochemical assays and histology. Again, all the equipment needed for doing this kind of research work is available in the center. We also have a dedicated a tool room where we have all the power drills and power tools for building or refining the tools or the prototypes which are done. We also have access to all the departments in the medical, in the engineering side as well, if we can't do something within our department. Next slide, please. So all this was built essentially as a place where people from all faculties of the universities can come in and converge and work together. And the centerpiece of our translational research program is what is called as the Idea Clinic. This is a open forum which we conduct once in 2 months, or even once a month if there is a demand for it. The idea is that we facilitate the collaboration between the engineering and basic sciences community with the medical fraternity. A lot of times what has been observed, or as probably you guys have also seen it or known about it, you have a problem which you need to be solved, and you don't know what is the exact technology which can be used. And on the other hand, the engineering side or the basic sciences people have come up with some solutions or devices or stuff which they don't know where it can be used or how it can be used in a more fruitful manner. This is a place when we come in, we have the engineering side and the basic scientists come in and brainstorm and come up with a solution or a possible solution or an approach to how we can solve this problem. We encourage and we invite proposals at all levels. The projects which are promising or which sound interesting, we actually should shortlist them, so to say, and then we support them with a seed grant. A financial support is given so that they can build that or take that idea forward. And this is a very fruitful session which we have usually, and some of the ideas which come out of it sound really crazy at times, but we have come up with some very interesting and innovative products out of this. Next slide, please. There are some ground rules, of course, during the Idea Clinic which we do. Next. There is no hierarchy, right? Once you enter the Idea Clinic, everybody is equal. He may be the senior most professor, or he could be the first year student who has just joined. Everybody has an equal say. No idea is stupid. Okay. Every revolutionary idea which came in sounded stupid at the very beginning, and so we do not consider any idea stupid. A lot of times our instinctive reaction as a senior person is like, oh my, that's a stupid idea. But no, we do not consider any idea stupid during this Idea Clinic. No is a big no, right? We never say no to anybody who proposes an idea. We never reject anybody outright. If the idea sounds or seems far-fetched at the beginning, we actually encourage the person to present again one more time by improving it. We give suggestions, or we work together with the person and try to get it done. And probably the most important thing in this is you have to leave your ego outside the room. Most scientists are temperamental, even more temperamental than medical practitioners. And if you walk in with a chip on your shoulder, nothing will come out of these sessions that we conduct. And so these are something which we fastidiously enforce during our Idea Clinics, and it has shown us some good results in the past few years. Next slide, please. So these are some of the just a rundown on some of the projects which came in through the Idea Clinic. One of this was actually this is a patented product eventually, and though we never took it to our product level, this was an improved design of the cold storage box for transferring the vaccines to remote locations. The student team worked they were from chemical engineering. They worked closely with the Department of Community Medicine, and then they improved the efficacy of the cold storage box by about 25%, which translates that in a normal time, if the vaccine carrier box holds the cold chain for about 10 hours, they extended it by about 2.5 hours, right? And so that is a good improvement in how much can be achieved through one of this. We had actually sponsored this project through our department. They not only won, it was published and presented in various forums, but it also got multiple awards nationally as well as internationally. Next slide, please. Again, this was a basic idea which had come through from the which was originated in the diabetic clinic. We proposed this idea to the students. They came up with an orthotic device with pressure sensors and a real-time data of how the pressure is felt in the insoles. These are insoles which can be worn inside the shoe, and we can then see the patterns of how the force is perceived. The figure on the left is a normal pattern of loading, which you see, but the figure on the right shows abnormal pattern where you see the red area shows increased loads, and so that is where the higher pressure is felt, and this is how we can detect or prevent diabetic foot ulcers if we know beforehand how the foot pressures have been changing over a period of time. Next slide, please. Uh, there was another proposal from our students to build a self-powered orthosis for stroke patients. This was a system which they built where there was no external power source and extremely lightweight and low cost, mainly used for rehabilitation, where you can train the person to use this device to strengthen his weakened side of the body. And this was validated in the physiotherapy department. This was done with mechanical engineering students and the physiotherapy department. So this is another project which has come out, which currently is in the phase of commercializing with some modification. Next slide, please. We also work very closely in the translational medicine and the translational research department with the orthopedics orthopedics department. We help in rapid prototyping and 3D modeling of extremely complex cases. We provide them with 3D models of the surgery or the surgical region that they are going to be operating on. This has now become commonplace. We were one of the first centers to start doing this in 2015. There are multiple commercial companies which have this as their business model, and so we still do this internally for our hospital patients as a service. Next slide, please. We've also been working with the School of Computing and their departments on creating AR/VR models for educational use. We create models for anatomy education. We've also been working with these Department of Psychiatry on coming up with ways for using AR/VR modules for psychological counseling and also for remote monitoring of physiotherapy patients, and to see how they follow their physical therapy routines and whether the rehab is happening in a manner which was expected in this case. So these are a couple of projects which have come out through the Idea Clinic. Next slide, please. Outcomes so far. So what have we achieved in the past 5 years? There are over 40 publications in international journals. I'm not counting the ones which were done in local conference papers and stuff like that. And this has been done in collaboration with the various departments of engineering and technology, as well as medical and health sciences. Dentistry, School of Dentistry is probably one of my most active collaborators in this endeavor of ours. We have about 5 book chapters written. We have had 2 textbook collaborations, which have already textbooks out in the market. Next slide, please. The ideas which we supported through the Idea Clinic, 5 of them have published patents based on the ideas which were encouraged and financially supported by us. We've had 2 companies come out of it. They have built their prototypes. One is an optometric product, and the other one is which has actually become a full-fledged startup, which is mentioned down here as Creator 3D. They are building a 3D bioprinter, which is essentially a 3D printer which can be used to 3D print organs or organoids. The big dream in the sky would be to be able to print actual organs in the future. However, right now that's a dream a little far away. But this is what it is. Brainwaves is another startup which came out. They've been using the AR modules which I spoke about for stroke rehabilitation. And so these are a couple of the products which have come and startups which have come out of the Idea Clinic which we have been running. Next slide, please. The way forward, we have to have multidisciplinary teams in all our research projects. Isolated excellence in silos, though it sounds great, we do not encourage it anymore. We do encourage all the research projects to have some collaborator across faculties. As you all may be aware, you may also seen it happen. And I mentioned before, scientists are temperamental. They do want to do things on their own alone, but that era is long over. Just like the first speaker, Dr. Thomas spoke about, that only about 17 to 18% of research actually reaches because reaches the common public, even though a lot of breakthroughs are made. Most of it is because it all happens in isolation, and unless you come together and take it forward, it's not going to work. Most of us used to be happy with just having a paper published or having a poster presentation somewhere. That is no longer the final endpoint of what we're going to be doing. We need to have patents, we need to make products, and then we need to be able to commercialize those products. How do we do this? How do we make this happen? You have to reward innovations and creativity at all levels. Timely and immediate recognition for all the people who come up with anything innovative or creative has to be done. Now with the social media with being all pervasive and everywhere, it is very easy to publicize and recognize your achievers in the university. Incentives have to be given at the university level. Could be financial, it could be a lesser coursework, more time for research and innovation. University taking up sending the creative or the more productive people for semester abroad, or go visit some university or industry locally or internationally. That could also be possible. Sabbaticals to rejuvenate and as well as to work hard on their products and take it to the next level. These are things which have to be done at the university level. This cannot be done just at a department level or college level. The whole management has to be behind this so that sorry. Next slide, please. I think I'm almost over.
Yes, sir.
Right. So that is my last slide. That's my email. If you guys, anybody has any question or wants to ask anything, feel free to write to me. If you guys have any questions, I'll be ready to answer. You can leave that last photo on. Oh, okay, that's fine.
Sir, should I continue presenting?
Nah, that's okay, fine. It's fine. the last photograph, you could keep it on the next slide.
Oh, sorry, yeah.
Yeah. If there are no questions, then I guess it's time for an early lunch break or whatever the next thing. I apologize for the not being able to present.
No issue.
Yeah.
So thank you very much for your valuable time, sir. So it is
Sure.
a very informative talk. So you elaborated how we should proceed from bench to bedside, and
Yeah.
also the various works done by the various institutes under the SRM. You know, a lot of research is going on in SRM, and even
Right.
the 3D modeling and 3D bioprinter, you are doing the work there.
Yes, sir. Yeah. So our first version has already been completed. Our department gave the students a grant of 1 lakh rupees in which they build their first prototype and proof of concept.
Okay.
With that first model, they actually won the National Innovation Contest. And so currently, AICTE has given them a grant of 40 lakhs to develop their second version to take it to commercialization. The first one only had a single nozzle printing facility.
Okay.
The current version has 3 nozzles for printing.
Okay.
And so we could have 3 different cell types with
Yeah.
3 different bioinks
Okay.
being printed at the same time. Right now, the focus, I think, though I would love at some point to be able to build cartilage or cartilage and tissue, cartilage and bone, osteochondral plugs or whatever. That is a little more complex thing. I hope to be achieving next year, maybe.
Bioprint is a very expensive machine. Region Q and
Yeah, correct. If you were to buy one right now, it's like somewhere in 60 to 80 lakhs, just like that. Like I said, the first one we built it in less than 1 lakh, and this product by the startup company, they're proposing to have it out in the market by February or March.
Oh.
But they are only targeting it around 10 to 12 lakh rupees.
Oh.
So it will be probably 1/5 or 1/6 the commercial price which is available, and most of these are foreign made products.
Yes.
So I'm hoping that it right now, you know, we're doing all the validations and all the fine testing, that someday we'll have it out as a full-fledged product outside.
Thank you, sir. Thank you very much for the information. And we will be contacting you for some doubts regarding this translation, how to proceed. We are also developing some of the tissue engineering products. But
Right.
How to proceed to the.
Sure.
From,
Definitely. Yeah. My email is with you. You can
yes.
call me anytime. Yeah.
Yes.
Thank you so much, guys.
Thank you.
So thank you, Dr. Shantanu, sir, for this very enlightening session, sir, on how to proceed in a university level and what all can be done. So thank you so much for that talk, and thank you, Nebu, sir, for once again moderating the sessions. Now we are actually 15 minutes early. Dr. Binsi, are you there? Ma'am?
Yes. I'm
Ma'am,
yeah.
are you ready to present?
Can you hear me?
Little
Am I
early?
audible?
Yes. Okay. Ma'am.
Yeah, no problem.
Okay.
Yeah, I can do.
Now I invite Dr. Subbalakshmi, Reader, Department of Periodontics, Pushperi College of Dental Science, to moderate the session.
Thank you. Thank you, Dr. Soumya. Good afternoon. And let me thank Team BioRadiance 2022 for giving me this opportunity to be here for moderating this session. And we've been having a wonderful the past 2 days of looking at what are all the different opportunities that we could do, which we come across in translational research. And next upon, I think we have somebody who's here to talk on a very, very pertinent topic, which is basically impediments, which would be there in the implementation of healthcare technologies developed from translational research. And for that, we have with us Dr. Vinci PK, who's a senior scientist with the ICAR Central Institute of Fisheries Technology, Cochin, Kerala. Welcome, ma'am. And we're looking forward to hearing from
Thank
you on
you.
the highlights on current impediments to measures and the measures to overcome them.
Thank you. Thank you very much for a wonderful welcoming note. I think I'm audible, right?
Yes, ma'am, you're audible and I can see your screen. If you could just change the slide, we could just confirm that's working too. Perfect.
Yeah. Is
Perfect.
it? Yeah. Okay.
Thank
Thank
you.
you. So, yeah, the topic is, as already mentioned, it's a very, very relevant topic, that is impediments in the implementation of healthcare technologies developed from translational research. And I represent the ICR Central Institute of Fisheries Technology with the headquarters at Cochin. And this is a ISO accredited and NABL accredited laboratory for referral as well as the reference lab. This is a small note about my institute. And regarding the topic, that is translational research, I was just wondering, like, how can I take the topic? Because it's a very vast topic, and I am actually standing at one end of this chain. Uh, where do I stand talking about this topic? So, what is my experience? I thought initially I was like, no, I am not the right person to talk about this one, because I'm basic scientist, I do basic research, and I'm not into much translational research, so I'm not the right person to talk about. But once I started learning or reading about the translational researchers and all, I felt like I should talk as a basic researcher, because I'm the starting point. I'm standing at the starting point of this one. So I should talk. I'm maybe the right person to talk about this one as well. So I'll just start about my journey into the translational research. Actually, we started the translational PR in collaboration with Pushpagiri Institute of Medical Sciences, and I'm happy, and I take this opportunity to thank the Pushpagiri Medical Society, as well as especially the Director of Research, Father Malvanjeri, Dr. Nebu and his team, for giving me the opportunity to step into this area of translation research. So, as I said, I started working on biomaterials, especially from marine resources, because drug from sea was, and now also it is a vast topic and the whole hot topic discussed everywhere. So I was so happy working on the biochemical aspects of certain biomolecules, especially biomaterials. So I was happily extracting these biomaterials, and I was characterizing this one. For years and years, I was doing the same thing. I was characterizing, and I was going for the commercial aspects of this one, because this our institute is basically a technology development institute. So commercialization is a major aspect, major mandate of the institute that has to be addressed. So I was characterizing, and I was developing several combinations of biomolecules, but initially it all went perfect. It was like well appreciated, but uh, after years, or means like uh, every review meeting, we have to present every annual review meeting. I was presenting the characterization parts. So as and when the years passed, the committee started asking me, how long you will sit characterizing the molecules, how long you will characterize the molecules, so what is the fate of these molecules, where it has reached, what is the fate of the fund that we have poured into all these researchers, what is the fate of those money that I have purchased the purest chemicals from, each one may be costing more than lakhs and all. So, what is the fate of all those chemicals? So, as a basic researcher, I was so confused how to take up, because I was looking at the product, end product, and the market. But I understood that moment that there is a lot of things to be done. It's not that easy to take the molecule to the market. There are, somebody told me that, yeah, I was doing prototyping that part. It was okay. IP was okay for me. But the next step on was I was totally stuck up because I have to go to the preclinical research. Then they said, yeah, this after preclinical research, the clinical relevance will be, assessed. Then it will be going to the clinical studies. Then after clinical studies only, I can start working on the pilot production and the full scale production or the scaling up for the industry. So I was totally stuck up with the middle components, that is the preclinical research and clinical studies. And in between, this has to be strongly supported by a funding agencies or funding from either from the institute or from the external source. I have to find the funding for this research, and above all, there is a lot of regulatory norms in between that is playing, and ultimately I may be getting a red flag or a green flag. It depends. So, 1 second. I think it's not moving, my slides. Is it moving?
No, now this one slide has not moved, ma'am. The one previously was working. Probably you want to stop sharing once and try doing it again.
Yeah. I'll do it. Yeah.
I guess
I
you
started
will.
with this.
No. I think you will have to stop sharing. Open the presentation mode and then start sharing, because right now we can see the other mode only.
How it is? It's not the full screen.
It's not the full screen. It is because I think you've shared the window of the PowerPoint. Yeah, it's not the full screen even now. So if you can,
Because it's full screen for me, right? Yeah. I'll stop sharing again.
yes, stop sharing. Make the presentation. Make it in the presentation mode and start sharing again. Or choose, any window. I mean,
I'll do. Yeah. Is it full screen now?
yes,
Is it full screen?
yeah, it is full screen now and moving also.
Oh,
All right.
yeah. Okay. So sorry for the disturbance. So this is the natural hydroxy. I started with natural hydroxyapatite. That I started working on natural hydroxyapatite since 2010. So natural hydroxyapatite that I extracted from fish scale as well as from bone, and the one which I wanted to take forward was the one isolated from fish scale. So I had different grades of hydroxyapatite. I standardized the protocol for the extraction of hydroxyapatite, and my prime aim was that the product should be available at cost-effective manner, and finally it should be available in cost-effective manner in the market. So we have our own protocol. We have standardized a protocol for extracting collagen as well as from the scale, and the final remnants will go to hydroxyapatite. So that protocol was standardized, and that itself took almost 2 years for me for standardizing that protocol. But as I said, I was talking about this one for years and years in all the committees about the natural hydroxyapatite. I knew that it should reach the medical field, but there are several hindrances or impediments in between. So what I did was immediately the product standardization extraction protocol, the one which was ready for the industrial scale, industrial hydroxyapatite grade, that was released immediately, and the technology was also commercialized to three parties, three companies. But still, I was not happy because I knew that it's a component, it's a composite of collagen as well as the hydroxyapatite that is present in the scale. So you can see here the fish scale, that is the microscopic view, that is inside what is there in the grid shape, it is hydroxyapatite, and that that is a basic component, inorganic phase of our bone, teeth, etc. So I think I need not discuss further about hydroxyapatite. There is one more presentation on hydroxyapatite, so in that one it will be discussed in detail. So I wanted to isolate both collagen as well as hydroxyapatite, and a process that is suitable for the best quality of collagen as well as the hydroxyapatite of medical grade had to be standardized. Then I started working on that aspect, and finally I could get hydroxyapatite that is suitable for the medical grade, that is with respect to its interconnectivity as well as its porosity, as well as the so many other properties had to be studied. That all I did. I think again there is a problem of not moving the slide.
Ma'am, are you there with us? I think she'll just rejoin us. I think she's lost her network. We'll just wait. I think Dr. Vinci has lost her connectivity. We'll just bear with her for a few minutes and she'll be joining us back.
Yeah, am I able to know?
Yes, ma'am, we're able to hear you now.
Sorry for in between. I think some issues with my. Yeah. Is it visible?
Yes, ma'am, it's visible and it's on full screen now.
Yeah, thank you. So this was the finally we have selected one grade which is specific to the biomedical biomedical specifications. So we opted one grade and we started our animal studies. This is all in as a collaboration with Pushpagiri Medical College with Dr. Nepu and team. We just moved on to. So this is the one which started with the immunocompatibility assay. Uh, the material was found to be suitable. Within 4 to 16 days, the tissue response could be stabilized and indicated by the fibrin network that is very much visible here. So
Ma'am, I think the screen sharing got stopped.
Yeah.
And do you want to mail us the PPT and do you
Yeah,
want to do that? Sure.
I'll do that. That will be the best. I'll do to
Ma'am, it's there in the text box. I'll just send it to you again. Ma'am, it's there in the chat box.
have you received? Have you received?
Not yet, ma'am. I'm just looking for it. Just give me a minute. Yeah. Yes, ma'am. We've got it. Just give me a time. Ma'am, you'll have to give us access for the same, ma'am.
Yeah, let me check. You have sent the mail?
Yes.
Got it?
Yes. Thank you. Just give me a couple of minutes. Let me just load it. It's just taking some time. Please do bear with us. Right. Ma'am, I'll share the screen now. Can you see the screen, ma'am?
Yeah. You can move the slide.
Ma'am, can you see the screen?
Yeah, I can.
Oh, all right, ma'am. So can we go ahead?
Yeah, please.
Sure. Please let me know when I need to change the slides.
Yeah, next slide. Please go to the next slide.
All right.
Next.
Thank you.
Next. Yeah, next. 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 Yeah, we were here.
Yeah, yes, ma'am, we were here.
Yeah. So subcutaneous implantation of fish-derived hydroxyapatite we tried, and it indicated good compatibility in terms of the period to be covered as well as the fibrin network production. So that can be visibly seen with HAP. It could be all the inflammatory responses were settled within 16 days of implantation. So next. This was further confirmed with histological. This is the same thing with blood vessels and fibrin network for subsequent fibrous tissue growth could be visible in all the animal studies. Next. Further, we tried for once it was found to be compatible. We moved on to the next part of the study, that is a maxillary bone regeneration. This was also one student by Dr. Nebu and team, and it was also considered for socket preservation. So in that experiment also we have extracted the maxillary tooth, and in one place, in one socket, we have placed the hydroxyapatite xenograft. Next. And the histological images also showed that the whole thing was actually compared against the osteograft, that is the commercially available osteograft, and the results were compared, and what we could find was the trabecular bone formation at alveolar margin and middle of socket was slower in osteograft, that is compared to the fish scale derived hydroxyapatite. So that was a highly promising observation for us because it was compared against the commercial one and it was giving the good performance. So next one. So this is better osteogenic properties also could be observed by the fish scale derived hydroxyapatite when compared to the osteograft. Commercially available osteograft. Next, that was observed within 8 weeks of period. So this is X-ray images of radiographic images of hydroxyapatite xenograft against the osteograft. Here also what we can see that osteograft at 4 weeks show traces of radiographically detectable calcification compared to the xenograft, but the calcification, extent of density of calcification was far higher in the xenograft, that is the FHAP, that is fish scale derived hydroxyapatite. In 4 weeks itself, it was highly visible and it was distinguishable compared to that of the commercial. So that result was confirmed with this image, and we just move to the next stage of the study, that is next. We have moved to the, initially we were using as a powder form, then we moved to the 3D printed fish scale hydroxyapatite bone implants as well, and its animal studies are going on. I think Dr. Nebu has got very good results with the hydroxyapatite 3D disc as well. So this was also giving a promising results to go ahead for preclinical evaluation. Next. Then alveolar bone regeneration also was compared against the bioglass. There also we could get comparable results because these are the candidate things, positive controls that we usually keep in bone regeneration studies. So we compared against the bioglass as well, and we could get that comparable activity of hydroxyapatite with bioglass. Next. That is the story with the hydroxyapatite, and we are still continuing its preclinical evaluation. I think the pharmacological department of Pushpagiri is taking care of those things, and as I said, we had several other components, products that was envisaged for biomedical applications, and another one such component was a cellular dermal matrix from fish scale. This you can see on towards the left side of that one, the top one, that was was the initial phase of the skin, that it contains a lot of pigments, it contains a lot of fat, lot of cellular matrices, gags, so many other things, which which are not preferred for a final refined cellular dermal matrix. So we had to work a lot on this one to selectively remove each component. Fat had to be removed, then pigment had to be removed, then nucleotides had to be selectively removed, gags had to be removed, elastin had to be removed. Like one component we had to selectively remove from the fish skin. And next, that you can see, this is the ventral surface of the skin where the collagen fibrils are oriented in a particular fashion. See, you can see it is all parallelly oriented. From that was the first stage. The fresh skin was like that, where lot of impurities, including meat, fat, slimy materials, everything was sticking onto that fish skin. Then each one we had removed selectively, that you can see that purification stage. Once it goes when it goes to the next stage, the thing that contaminants were selectively removed. Towards the fifth stage, you can see at least the fibrils are very neatly and clearly are oriented in a vertical and a parallel manner. Next. So this was the final one which we got after refining all the series of selective removal of the component. This was the final one which we got as a cellular matrix, and we confirmed the purity of this cellular matrix by redissolving in a solvent, that is a collagen solvent. We redissolved it. We got a very fine and viscous collagen solution without any settlement. That is very much important, because if there is any elastin and other component, that won't be soluble, so it will be settled down. So all those things had to be made sure. Then we solubilized it into a very uniform solution, viscous solution, and again confirmed the process by cross-linking the collagen. So once you got towards the right is the cross-link collagen, that is in the mold shape. What you can see is a cross-link collagen. So the purity was as the purity of that collagen had to be ascertained with various analytical techniques that was followed, and finally we confirmed the collagen in the pure form. So this cellular matrix was used for the further wound healing as well as the bone regeneration matrix, as well as the collagen extracted could be either you can cast it as a membrane, or the acellular dermal matrix can be used as a. So we have the option of both. You have acellular matrix in one hand, or you extract collagen, or you simply dissolve in a solvent and you cast it on to a membrane shape. So these both options were made open, because once again, because as a basic researcher, my focus should be the ultimate product. The ultimate product should be available in the market in a very cheap, or it should be affordable to the large. So with that intention, I have made two options. One as such you can use, or one can be melted or can be redissolved and can be made in the composite form with other biopolymers and can be recasted again. Next. Now the thing is that the fish matrix, you can see what is available. The commercial product in the market is the Heligate, as the one we have taken as a candidate commercial counterpart. So we just compared the structure, microstructure of Heligite as against that of the fish skin matrix. You can see fish skin has retained its, or the collagen has retained its original nature. The native structure of collagen was retained. That means it's very much ideal for to act as a natural substratum to mimic our skin. So where the collagen is oriented in a very parallel manner as bundle collagen fibrils. Towards the right, you can see that is the Heligite. You can see it's like a cross-linked and recasted one. Collagen was dissolved in some solvent, and it was recasted. So it has lost its identity. Total fibrillar identity was lost, and it was giving the microporosity, but the total integrity or the original nativity of the collagen was lost. This was confirmed in the, next slide. This you can see, this was confirmed the FTIR profile where many of the peaks were missing. In the collagen, the native collagen, what all signatory peaks were there, all those peaks were missing in the helix. That means that those functional groups have been lost during the process of the casting process, or it has reacted either with the solvent or the other material that has been added to that. Probably the plasticizers and all they might have added, probably that has reacted with the functional groups of collagen, and it has lost the native structure of the, or the native activity of the collagen. So that was very much visible in the FTIR, where the original, all the native signatory bands of collagen was visible in our fish skin derived cellular matrix. That was a promising thing for us to go forward. The next slide. In vitro cell viability was also assessed as the basic one, or the preliminary step. What we could see that cell attachment assay gave better results for fish skin derived dermal matrix. That you can see the differences in the intensity of this one. So 100% of fish skin extract showed cell viability. That was also confirmed. Next. So in vivo biocompatibility, as usual, we went for this one. You can see the difference, the fibrillar response in in second week itself. We could find that it is settling the immune response, because that is a major thing that we all are worrying about, whether the body will be rejecting the matrix, whether immune response will be beyond the limit, all those things, because that shows the extent of purity of the matrix. If something extra is there, definitely that will be shown during our once it is implanted in the body. So fibrillar network formation, that was that itself was a promising one. Next. This is the fourth week. You can see intense network formation in fish skin when compared to the Healy guide. So this is also done by Dr. Nebu and his team. Next. Another one, so that part is also, we are just moving ahead, moving forward with the fish skin derived cellular dermal matrices. That study is progressing. Then another one is the demineralized fish bone. So here what we did was, this is all the thing what I wanted to tell again, because all these things were there with me, lying with me for years and years, and I was just kept on characterizing the thing. So finally, when Dr. Nebu and the team joined with us in taking forward only all these things, happen to the translational mode. So demineralized fish bone is another promising one, where bone is also, bone is composed of collagen and hydroxyapatite. So in the first one, I was extracting hydroxyapatite, I was removing collagen. So in the here, what I did was, I removed hydroxyapatite and retained collagen. So collagen network was, that is congenial for the calcification was made, and the same thing, it was compared against bioglass and the commercial counterpart, and the major thing was the degradation rate, that the biodegradability was a major question, and we could get a better results for this, comparable results for demineralized bone as well when compared to the bioglass. Next. MTT assay, as usual, we did, and that was also giving promising results, and 80 to 120 micrograms of fish bone was found to be optimum to induce cell proliferation. Next. I'm just moving fast because the real topic comes towards the end. So before that, I have to talk about where we stand in translational research. So wound healing and regeneration properties of the demineralized fish bone was also confirmed. Next. in vivo biocompatibility or demineralized fish bone was also assessed, and we could find a compatibility histopathology after 24 days. We could find that the third week onwards, no foreign body reaction visible or any chronic inflammation post implantation, and the demineralized fish bone got integrated into the tissue. So new blood vessels started appearing. So that was also found to be biocompatible without any contaminants that is responsible for the implant rejection. Next. So this one, again, we have tried for the cranial bone regeneration. Next. The CBC assay image was taken, and you can see that between this one, the fish group T test group B was giving higher intensity. Next. Next. Why it has gone back?
Yes, ma'am. I'll just.
It has gone back, I think.
I'll just minimize it and check, ma'am. I think those slides are repeated again, ma'am. I'll get that.
Towards the top.
Ma'am, if you could specify the slide number.
Yeah. Please go behind. Yeah, towards first. No, before that. Before. No, down. There, I think it got mixed up. Right?
Yes, ma'am. From this slide, I think again it's gone back to the first few slides.
Okay. Towards down.
Ma'am, is this the slide?
No, previous one. Previous ones. Previous. Yeah. Next one you can give. Yeah, that one. Yes.
All right.
Another one. Yeah, this is good enough. Yeah. Another one, the GTR scaffold, that is with hydroxyapatite and titanium oxide nanoparticles. This was also conducted at Pushpagiri. So we have incorporated titanium dioxide, and the combination was optimized by taking different concentration that is congenial for giving a homogeneous pore size distribution, because that was the major thing. Once we incorporate this one hydroxyapatite, it won't be easily soluble in that one, but it will be settle down, and it will make the scaffold more porous. So that was the major challenge in this one, and we could finally standardize the material. And the next one, again, osteoblast cell attachment studies also was analyzed, and that shows that it is attaching more, and it is congenial for the attachment. And when compared to the Healy-Guide, again, we were keeping Healy-Guide as reference material, and it was giving a better results compared to.
Ma'am, you're audible, ma'am.
Yeah. Next one. Yeah, degradation rate and as well as the cytotoxicity assay was also analyzed. Somebody else also.
Yes, ma'am. We'll just fix it, ma'am. Yes, ma'am. You can go ahead.
Yeah, the next one. Subcutaneous tissue compatibility assessed again, and it was shown that by 4 weeks post-transplantation, the immune responses evident during first and second weeks has completely disappeared, and the tissue appeared normal. So that was also confirmed. Yeah, next. Hemostatic and bone healing chitosan scaffold was another one that we attempted together. We prepared a scaffold of chitosan that was mainly intended for wound healing and subsequently for bone regeneration applications was intended. Next. That was also studied for the compatibility studies, and By 12 days, inflammatory reactions were settled and then collagen network was formed. Next. These were some of the things that were directly involved in the biomaterial that was directly involved in the tissue engineering. But there are certain important things that we all know, that is the growth factors. So we have some more biomolecules that has been isolated from marine resources that is having some cell regeneration properties, and one among them is the astaxanthin shrimp head oil complex that has been extracted from shrimp head oil. So this is our product that is, you can see that the marketability of this product is also very high, that is 10 rupees per tablet is the cost. And hydroxyapatite, I forgot to mention, that is also like less than 200 rupees per kg. So that the one that is available in the market, that the synthetic one that is available in the market, that is per gram it costs around 1,000 rupees. So you just see the marketability of the material that is available, fish skin, fish bone, and all. It is easily available in our country, the tropical country like India. This is the resources abundantly available, and it is just nearby our markets itself. It has been accumulated. So only thing is that we should have a systematic approach for collection of these materials and extraction of these biomaterials from these base. This is basically base components. So ultimately, the major thing that is about affordability in the market will be satisfied. So such researchers in the basic research aspect is very much necessitating one, and the CFT was working for that aspect for years, and now we are moving more into the translational aspect. So this is one among them, that is astaxanthin. Next one, its properties are also discussed in this one. So cell regeneration properties are significantly higher when astaxanthin was incorporated. So next one, you can see the anti-inflammatory property of astaxanthin. So that is also significantly reduced when astaxanthin was incorporated. Next, anti-tumor activity, that was also well established. Next, HeLa cells, that is also cervical cancer. The other one was the osteoblast. Then now this one is the cervical cancer cell line. Also HeLa cell, also cell proliferation could be arrested significantly, about 60 microgram. You know that 60 microgram is a very small quantity. That is 10 rupees per tablet. When I say that contains almost 1 gram tablet. So 60 microgram, the cost wise it is very less. So there are, the only thing that we should remember is there are many cheaper candidates for us, that is from the natural resource itself, that has to be identified, and it has to be put to channelized to the translational research area, and finally the product should be available in the market at affordable price. So next one, cytotoxicity was also confirmed that it is not at all it was satisfying all the norms, and it was not toxic. At the same time, it was showing a wonderful property of focused targeting of cancer cells, because it was, as such, it was to the normal cell, it was not toxic, or it was not killing the cells, but it was targeting the cancer cells. There was a significant reduction in the cancer cells. So that is a promising area for further exploration. So next one, marine melanin is another candidate. That is, you may be knowing that squid ink, that is the ink contains an important biomolecule, that is the melanin, that was also subjected to some basic research. So what we found, that it is also having cell proliferation effects. It's a wonderful UV protector, UV absorber, that we all know, that is the melanin is basically doing that function of UV absorption, but it is at the same time it is a wonderful cell regeneration. It is showing cell proliferation activity as well. So next one. So collagen peptide, as I initially mentioned, the collagen can be hydrolyzed into fine powder. So collagen as such may be used for the membrane fabrication, material fabrication, but collagen peptide as such is a growth inducing or the cell proliferation activities which showing very good cell proliferation activity it is showing. So it can be used as ideal candidate for cell regeneration studies, and it can be incorporated. The very technical advantage of a collagen peptide is that it is very well soluble in water, so it can be easily incorporated into any material, to any form. Can be in gel form, it can be in solid form, can be in film form or paste form, any form it can be easily incorporated. So that part is also our under study is going on. Next. Another thing is the bioactivity is active extract from marine microalgae, that is nano. There are several candidates. I have just indicated just one organism, that is Nanochloropsis occulata. So this was also carried out, and it was found that it is having beautiful cell regeneration properties. At the same time, anti-cancer and anti-inflammatory properties were also observed. So this is also, we are escalating the possibilities of seaweeds as well as the microalgae to be used as growth enhancer or the tissue proliferation matrices. So next one. Here comes the major hurdle. As I said, these are the major hurdles that as a basic researcher that I face. So I just went through all the things, like initially I was so confused, like what to start with, what to know, whom to approach, what are the regulatory bodies, where all I should apply. All those confusions will be there as a basic researcher. So the first hurdle that I faced was, I thought I'll share my experience first rather than going for the real in-depth translational research. So as a basic researcher, the very first thing which I faced was, this is basically a multidisciplinary approach. So that has to be followed, that's very sure. So there should be a basic researcher, there should be a translator research from that area, but probably the basic researcher will be from totally different area. Like me, I am not from the medical background, I am from biomolecular extraction or first product design part. So the next comes the medical field. So there was a total differences in the discipline itself. So it is totally, it's not only multidisciplinary, there will be interdisciplinary within the division, within the department also. We should have the interaction. So that was one of the major challenge that I faced, because whenever I approached several medical colleges and during my initial phase. So maybe like they have their own restrictions. Probably clinical research, they were more into clinical aspects. The translational research is not that much mandatory for them as per their maybe like the institutional mandates. Translational research was given very least importance, but probably that's the thing. Or maybe like the clinicians, so they have lack of time, because they are not more, they are more into clinical aspect, they don't want to spend more time for the research aspect. So that is one of the things. Then the most important thing is to approach the right person, who right investigator, the right translator researcher, who is working on the given aspect, on the particular subject. So shortage of translational investigators, that too subject specific investigator, that is one of the inference. Lack of institutional support. Fortunately, I had institutional support, but that may not be the situation for many. CFT was asking me to go for translational research, so I got all the support from the institute. So many research institute, the translational research is not as part of the scorecard, or may not be as part of their mandate. So they may not support the investigator or the scientist going behind the translational research. So being a technology institute, we wanted the product to be launched in the market. So translational research, it's mandatory for us to do. So in that way, I got support from the institute, but that may not be the situation for many. Then inadequate financial support. The financial support is very much in unhindered support is very much required for the translational research, because we know that it is very much investment intensive business. It has, once it starts, it has to be finished till the point, the finishing point. You should have unhindered support either from your institute or from the financial bodies. So that uncertainty in funding pattern, so that is also one of the impediments. Then lack of infrastructure. So many of the institutes who does basic research, they may not be having the infrastructure to proceed into the translational research. The only thing what they have to do is like that, approaching the translational researchers or the other institute where a well-developed infrastructure facility is available. So that is also one of the major impediments that we have found. The next one, insufficient research recognition. Whether you do basic research or translational research, what you are ultimately getting is one and the same. You are not getting higher recognition out of translational research than the one who is doing basic research. Sometimes the basic researchers may be getting published in a very good reputed journal than the translational research. That is also true. So people are more happy, we're very much satisfied with the basic research, the outputs that we get from the basic research, and the recognition that is we are getting from the basic research. And translational research, it takes time, it takes effort, it takes your inter collaboration, inter laboratory collaboration, interdisciplinary collaboration, multidisciplinary collaboration, everything. So people are not very much interested to put into that area. They are very much happy with the one, unless they are getting an additional benefit out of translational research. So that is one of the major points to be considered in research as well as R&D structures. Then absence of government consortium with different stakeholders, institute, hospitals, and regulators, because this is one of the major things that national wise it has to be considered. That is, there should be a common consortium involved, because this is a multidisciplinary. There should be a common platform where all the discussions should be done on a single platform. So that is probably in India, that is one of the things. Then insufficient communication between researchers and stakeholders, because stakeholders, major industry stakeholders, they are lying at the end of the chain. They are nowhere in between. The basic researchers at the one end, and the translational researchers, they are filling the gap, and the industry partners, they are lying at one end of the chain. So there should be proper communication between the basic researcher itself. Then from himself, the communication gap should be sealed. So a long time period for completion, that is another thing, because we know the clinical studies are not that easy, and it cannot be, it takes a very huge time. So increasing regulatory burden, above and all, there are several regulatory norms that may be keeping on changing, and that has to be, that is also reducing, because that reforms may be for reducing the harm to the participants, or protection of privacy rights, data management, financial conflicts. So many factors are playing the next. So in brief, the potential is for translational discoveries should come from a strong product-oriented and evidence-based basic research, which can be validated in the field. The funding agencies need to redefine the policies, strategies, and formulate the guidelines to support fundamental research by bridging the translational gaps. Then regulatory reforms should be implemented so as to facilitate early approvals. The patent laws need to be restructured as to avoid delay in translation research, and the product can be launched in market at affordable rates. The conceptualization of TR programs should be in close association and consultation with the stakeholders in a competent consortium. Next. So the very beginning, we are hearing the slogan of TR. translation research, that is bench to bedside and bedside to community. That is very much possible only by bringing all interdisciplinary stakeholders on a common platform. That is the first step. And towards the end, we have introducing the product in a cost-effective manner. The product is, it has to be put in, inverted comma. The product means with respect to all the things. That means it has to pass through all the norms of translational research. Then only the product will be there. But it has to be available in a cost-efficient manner. So there is a huge gap between the first thing, interdisciplinary stakeholders platform, to the product. That's a huge gap. So how effectively we can fill this gap? The success of our slogan, that is bench to bedside and bedside to community, is possible only with that sort of approach. Yeah, next one. Thank you. So these are the gist of the technologies that we had developed in CIFT and in collaboration with Pushpagiri, and some of the things, some of the impediments that we have faced during our journey was discussed. Yeah, thank you. If there is any question, I'm happy to answer.
Thank you, ma'am. That was an interesting session. And thank you for taking us through your translational research journey in detail and discussing all the hurdles that you've met right from the beginning. Now the session is open for questions. If there are any. I do not see any questions, ma'am. Thank you so much for joining us today and for all the work that you've been doing with us. Thank you.
Thank you. Thank you for giving me this opportunity to interact with you all. Thank you very much.
Thank you.
Thank you, Subhlakshmi ma'am. Thank you, Dr. Vinci ma'am, for this session. Now we are moving on to the lunch break. Prior to the lunch break, I invite Dr. Elizabeth, HOD of Department of Pediatric Dentistry to Pushperi College of Dental Sciences, to announce the results of the poster presentation that was held yesterday.
Good afternoon to everybody. I'm privileged to announce the prizes of the poster presentation, which was presented by most of our PG students and staff who were taking. So shall I start from the third prize? The third prize is awarded to Mridura Sridharan of Mahatma Gandhi University of Kotte. And the topic was nanocellulose composite for biomedical applications. So congratulations, Mridura Sridharan. The second prize is shared by 3 posters. For the 3 include Dr. Vinci U of the Department of Periodontics of Sri Shankara Dental College, Varkala, and Dr. Netra Narayanan, Dr. Aditya Sunil, and Dr. Manasa Anil from the Department of Periodontics of Mar Vesalius Dental College, and Dr. Sheena S. Raj of the Department of Conservative Dentistry of Pushperi College of Dental Sciences. Congratulations, all 3 posters and those who participated. And now coming to the first prize. The first prize goes to Dr. Vimal Thomas Uman of the Department of Periodontics of Pushperi College of Dental Sciences. And his topic was in vitro biocompatible assessment of chitosan hydrogel loaded with solution gel synthesized bioglass. Congratulations, Dr. Vimal Thomas. So thank you.
Thank you, Elizabeth ma'am, for announcing the results. Congratulations to all the winners. Now we break for lunch, and please do rejoin by around 12:50 for the next session, which will begin at 1 o'clock. Thank you.
Good afternoon. Welcome back post lunch. We begin the next session of today's workshop. It's my pleasure to welcome Dr. Sachin J. Shenoy, scientist, laboratory animal veterinarian at the Sri Chitra Tirunal Institute for Medical Sciences and Technology, Tirunelveli. Sir, handing over the session to moderate the next session, sir. Handing over the session to you. Sir, would you unmute yourself, please?
Yes, like hope I am audible.
Yes, sir, you're audible, sir.
Yeah. First of all, I'm thanking the organizers for considering me as a moderator for this session. I think we can introduce the speaker now, madam.
Yes, sir. Yes, sir. You may. Please.
Yeah. So the next speaker is Dr. Mohammed Ashraf P. He's a principal scientist, Material Science and Environment Fishing Technology Division, ICAR, Central Institute of Fisheries Technology, Cochin, Kerala. He will be talking on the topic development, synthesis, and validation of natural hydroxyapatite. I think he will be covering various sources of natural hydroxyapatite, methods of synthesis, and characterization. Over to you, sir. I think sir should unmute. Kindly unmute, sir.
Sir, are you not able to unmute, sir? Sir, we have made you the co-host here. Sir, would you try once again?
Are you hearing?
Yes, sir, you're audible, sir.
Okay, fine. Thank you. This one I'll proceed. Basically, I have to make it a window.
Yes, sir, you can share your entire screen. I think that would be better, sir.
Is it? Okay, okay. 1 minute, just. Entire screen, okay.
Yes, sir.
Then how I can go to my presentation?
Sir, you may, if you have minimized your presentation, you can maximize it and put it on the presenter view, sir.
Okay, 1 minute, 1 minute. Okay. Is it visible?
your screen is not visible, sir. Are you sharing your screen now?
Yeah, I'm trying to share. Tom wants to share the contents of your screen with me. Yes. It's not sharing. One minute.
Sir, you're not able to share your screen, sir.
Yeah, I
Sir,
could not
would you-
able to.
Sir, would you, if you can mail your PPT to
Yeah, okay, I'll make it.
the mail ID that is there in the chat box, sir, we can share it from here, sir, from our side.
I'll just 1 minute. What is the mail ID, please?
meet@pimsrc.edu.in. It's there in the chat box, sir.
No, it is. I could not see anything there. Okay, 1 minute.
Sir, are you able to see the chat box now?
Now I can see.
Yes.
Have sent already. Got it?
Sir, is it sent from your side?
Yeah, I have sent already.
Sir, we have received it. A few minutes, sir. We'll just get that.
Okay, fine.
Sir, is the screen
Yes,
visible?
it's visible. It's fine. Good afternoon. Thanks for the nice introduction, and I'm very thankful to the organizers for inviting me for this giving this presentation. Myself, Muhammad Ashraf, I'm working as a principal scientist in Fishing Technology Division of ICAR Central Institute of Fisheries Technology, and I'm the development, synthesis, and HAP in natural content is I'm talking about that. My talk next. My talk comprise of, I'll talk about little hydroxyapatite, its synthesis and its sources, and the methods to test, and the fish phase methodologies, etc. Next, please. Then hydroxyapatite is calcium phosphate similar to the human heart tissue morphology and its composition. It has hexagonal structure and is generally the stoichiometric ratio of calcium phosphate was around 1.67. And next, please. And generally, thermodynamically, hydroxyapatite is the most stable calcium phosphate compound under physiological conditions in respective temperature, pH, and the composition of the body fluids, etc. And this is the general IUPAC name. Pentacalcium hydroxide triphosphate is the name. And these are all the details under IUPAC system. And next, please. And then the ratio between the calcium and phosphorus found to be the most effective in bond regeneration. And natural HOP is most of the time it is not very constant. 1.67, it may 1.5, and it varied from 2 up to 2, and all depends on the materials. Generally, the natural that is what the calcium position, because of this was due to the calcium positions are occupied by sodium, magnesium 2+, aluminum, or etc. And next, please. Sometimes this phosphate positions also occupied by hydroxyl ions and fluoride ions, and this may be the reason it is getting this ratio is changed between 1.67 to less than or higher. And generally, the natural hydroxyapatite is contaminated with trace elements, but it is most of the time it was explained that it is beneficial for it promotes the tissue bond regeneration purpose in that. And the presence of 1 to 10% of the strontium ion, it was Cappuccini et al. In 2010-8 told that it is 1 to 10% of the strontium ion in synthetic hydroxyapatite enhanced osteoblastic activity and differentiation, and also inhibited osteoblast proliferation and production. Next, please. Generally, synthesis of hydroxyapatite in chemical methods, salt gel synthesis. Generally, for salt gel preparation, it is generally used by calcium nitrate, phosphoric acid, and it becomes and different temperature, all those hydroxyapatite generally, or diammonium hydrogen phosphate. Then what is the method used for electrochemical system? Generally, EDTA, calcium phosphate as an electrolyte is used for used in between the carbon electrode, and this way also it is generally used. A lot of literatures were available, the synthesis of hydroxyapatite and its attachment over the metal systems. So this is also one of the important method for preparing hydroxyapatite coating over metal implants. And another method is hydrothermal synthesis. Generally, hydrothermal synthesis means it is a steel vessel lined with Teflon, and it is a closed system. If you can heat it up to Teflon means it is around 250 degrees centigrade, or it is varied from depending on the system. So we will get the varied morphology compounds, such as nanorods, microspheres, external prisms. Different depends on the temperature and material and what are the systems we are using. Different structures were obtained in hydrothermal. So it is considered as one of the green system for synthesis concerned. Next, please. And generally, I'm talking about the general hydroxyapatite from natural sources. For example, bone, fish bone or fish scale, clam, eggshell. These are all the rich in calcium phosphates, and they are crushed into smaller sizes, and it is washed with water or boiling water. Depends on the different. So many methodologies are available. Just I put it in a nutshell. It is washed with water, boiling water, or boiling water plus acetone and chloroform. So different surfactants, alkali, alternatively, or whatever it is. So much methods are there to remove the fat, protein, and other components present in the bone tissues or clam shell, etc. So after that, what they will do is they will put it into this material in 700. Different literature says that it varied from 700 to 1400 degrees centigrade. So it depends on the different authors. It is varied. It is so hydroxyapatite is formed 700 to 1400 degrees centigrade. And also it is reported that greater than 800 degrees centigrade, if you are using the beta, there is a possibility of beta tricalcium phosphate also along with the hydroxyapatite from the natural materials. Next, please. And these are all the natural materials of natural hydroxyapatite. For example, mammalian, different methods. It is already described. I told in general. These are all the methods: calcination, alkaline hydrolysis, hydrothermal, and then hydrothermal combination methods, and microwave irradiation, pyrolysis. Different depends on the minerals and materials. This Puad et al. Has already explained in detail in his review on 2019 regarding different materials used for methods used for preparation of natural hydroxyapatite from the natural materials. Next, please. So this advent of nanotechnology, the major impact is major impact has created in the material science field, and production of nanomaterial has gained considerable importance because the size is small and we have to use very less amount in the system instead of using higher amounts. And nano hydroxyapatite also we can prepare from natural materials, and it is very interested in the biomaterial side, and its use in prosthetic application. Similarity in size, crystallography, chemical composition with human heart tissues, bone and teeth enamel are largely composed of these materials. So nano hydroxyapatite has got very much importance. I will come to that. Next, please. And what are the important characteristics of this nano hydroxyapatite? Is highly biocompatible, bioactive, and osteoconductivity is very high, and then non-toxicity and non-inflammatory nature. So I'm not talking about much applications. You are all knows because you are all done this dental doctors, and this bone tissue engineering, bone void, fillers, maxillofacial, dental surgery, restoration of periodontal defects. Next, please. Endodontic material like pulp capping, repair of mechanical furcation, perforations, apical barrier, etc. And then early caries, lesion treatment, drug and gene delivery. These are all some of the applications using hydroxyapatite, nano hydroxyapatite. Next, please. Next, I will talk about, we have a small work we have carried out in using fish scale. Fish scale is generally seen in all fishes we have, and we have taken a fish from that Katla rohu, and we have prepared nano hydroxyapatite and carbon dots from this. Next, please. And generally, we know that global fish waste is 18 to 30 million tons from the seafood industry, of which 8 to 10% is fish scale. So the availability is very high, and then the fish scales are generally it is thrown out without much without much any utilization. So the this fish scale it contain nano hydroxyapatites are aligned in regular matter along with the collagenous materials. This composition provides higher strength, resilience, and pressures, enhanced osteoblast differentiation, osteoconductivity. Fish scale is abundantly and cheaply available material, and natural HAP is derived from biological sources are preferred over. Then, as I told you, it is synthetic counterpart. Next, please. And this is the structure of the fish scale. It contains, there are 3 layers. One is called osseous layer, then a transition layer, and second one is matrix layer. This osseous layer is the topmost layer. It contains hydroxyapatite fibers and interacts with the biological material. And then a transition and this matrix layer, the second layer, this one. So this layer also you can see that it contains collagenous protein along with the calcium, collagenous material with the protein arranged in such a, and it is pinned with the calcium materials, and it is like a plywood-like structure, and it is woven with the calcium compounds. Next, please. Generally, what is the method generally used? Hydroxyapatite was prepared from the fish scale, actually, earlier also. But what they will do is, they will undergo a lot of washing and all. So this collagenous material will be lost. So you can prepare either collagenous material or hydroxyapatite. You can. So what we are telling is, instead of this collagenous material, also we can utilize some utilized for producing a new product that is called carbon dots, and this hydroxyapatite also, along with it, we can extract it, as I told you. And the crystalline properties are comparable to synthetic analog. So Barkha theta or synthesized in hydroxyapatite, nanorods, nanoflakes, and pure form respectively in thermal processing at 750 degrees centigrade, at holding to subcritical water flux, like that. Different methodologies were used for preparation of hydroxyapatite from natural, and needs to extract collagen, then hydroxy ideally requires more acid. So if you are using this taken out for collagen peptide from this one, you need a lot of acids were used. So it is all along, it is not very natural, because ecologically good material. Next, please. Method, sorry. And then synthesis of hydroxyapatite we have carried out in hydrothermal methods. So in that procedure, what we have used is acetic acid was used as a medium, and then this shell scale was used as a raw material, and then this all collagens were converted into this nanocarbon dots, and then the other hydroxyapatite was crystalline in the crystalline form. It is available along with the filter paper. So that we have this. So I will come into that. Next, please. This is the methodology we are following: fish scale, and then we have got carbon dot with a fluorescent material, and then we have prepared a biorefinery like system. That is what we have done is hydroxyapatite we have synthesized from after sindering the precipitate obtained after filtration of this one. And what we have seen is that along with the acetic acid residue, carbon dot, we have seen a crystal, white crystals. So these crystals were, while we have evaporated to dryness, it becomes formed, it has become more visible, and these crystals were extracted out. And then what we have done is, again, we have heated to 800,000 cinder to 1000 degrees centigrade, 800 degrees centigrade. We have got beta dicalcium phosphate. Next, please. And these are the carbon. This is the carbon dot. This is a highly fluorescent material. This, by seeing it, is a brown color solution, and we can see this is a UV visible spectroscopy measurements. We can see two peaks. The first peak from this blue line is indicated that pi-pi transition. That is because of the double bond. Generally, carbon dot is formed with a double bond, that conjugated double bond system, and then the other groups were represented this second peak at 270, like that. Next, please. And this is the morphology of carbon dots, and it is having very 25 less than 4 to 5 nanometer size, and FM topography also. You can see there is a small variation in the sizes between, because it may undergo some type of agglomeration during analysis. Please, next. And this is natural. What is the advantage of this prepared nanohydroxyapatite from fish scale? This hydrothermal method is purely white crystalline powder, and with excellent color. Compared generally, what we are seeing is the hydroxyapatite having some off-white color if we are preparing through direct sintering of fish scale or after washing it up. And we have got an yield of 6.7% per 7%, and it is UV visible spectra. We have seen different major peaks in the 280, 335, and 384, and we can see that some of the peaks were missed from the spectrum. That indicate that this whole shell is the scale is converted to different products, and this hydroxyapatite is one among them. Next, please. And the FTIR spectroscopy studies, we have seen that major absorption. You can see a tower-like peak in this area, the 650 to 1100 degrees, 1100 nanometer centimeter inverse, because the major peaks were seen in 940, 970, and then 1021, 1021, 114. What it is shows that it is mainly due to the PO system, symmetric stretching of PO system, and then G3, G1, G3, different bending, all spectral signatures are assigned by different notations, that is G1, G2, G3, etc. And we have seen some OH vibrations also. Phosphate was 665 and 3545, 42. But we have seen that type A system is containing very little amount of carbonate in traces, and it is that way has appeared in the this also reported with Maiduniac also. It is after replacing hydroxyl groups. Next, please. Then FTIR studies also showed that most of the peaks were comparable to the ICDD cards of 090432, and the typical peak of hydroxyapatite was 31.63. It is just comparable to the original one. It is because some variation because of the hydroxyapatite is standard hydroxyapatite, because it is derived from natural material. And it may also have influenced the temperatures, organic molecules present in the carbonate in the system, and because the carbon dioxide, while heating, it will go carbon dioxide in water and leaving behind the crystalline and finer nano-sized hydroxyapatite particles. Next, please. And this is the atomic force microscope studies and TEM studies. We can see that the hydroxyapatite generally always seen as a cluster form, and it is we can see the SAED interplanar distance was 9.39 and 5.8 nanometers, and it is very XRD data calculations also we have seen is 6 to 8 nanometers is the size of the material, and AFM evaluation showed that is less than 25 nanometers, around 25 nanometers is the size. Please, next. And then after that, what we have got hydroxyapatite, and then what we have seen that this nanocarbon dot solution, we just evaporated dryness. We could see a lot of crystals were seen in the bottom. Then we have extracted the carbon dot separately, and then these crystals were filtered out, and we have analyzed its characteristics by X-ray diffraction procedures, and then we have seen it is a off-white powder, and it is also a nanostructured material, and this is where this X-ray diffraction was compared with ICSD 14313, indicating the formation of beta dicalcium diphosphate. So in this, we have got, apart from the hydroxyapatite, another form of calcium form that is called beta dicalcium diphosphate. Next, please. And this is FTR spectra. You can see the difference in FTR spectra of hydroxyapatite and this beta dicalcium phosphate. It is having sharp absorptions 1155, 1055, and so on. So definitely it is different from this hydroxyapatite. As are most of the peaks were due to the PO vibration, pyrophosphates, and HPO4 ion, and P2O7 peaks and all. So it is a mixture of different compounds, please. Next. And this is the AFM mix. Similar to that, it is 28 to 35 nanometer was the size. And okay, next. And what is the mechanism when we put it in a hydrothermal reactor along with acetic acid? This shell, these scales were when put it in a higher temperature, it becomes some relaxation is happened. This is the mechanism is happened at 180 degrees centigrade. this when some type of relaxation because of the presence of acetic acid, then this acetic acid is very notorious to interact with the calcium salts. So what they will do is it react with the calcium salts present in the scale, and then it started degrading, and then it degrade the collagen also because of the presence of first it will cut the calcium link, and then the collagen will be freed from the scale, and then there are two components, and then hydroxyapatite and the calcium salts, and then the collagen. This collagen were easily converted into undergo, because it is released from the system, then it undergo reorientation and form carbon dots, a fluorescent material, and then this other part is hydroxyapatite, and then dicalcium diphosphate was coming along with the CD. Okay, next please. And this is the summary. This is the summary of flow chart. We have carried out this work, and it is completely a bioreactive type of synthesis, and it is having a lot of industrial importance. Please, next. Next, please. Then with this carbon dot hydroxyapatite composite was prepared, and is a fine brown powder, and it also has still we are working on its application. Please, next, please. And another material we have at present we are doing is the carbon dot collagen peptide hydroxyapatite composite for especially for bond regeneration purposes. Some of the samples we have already given to my friend, and work is going on. And what we have seen that we have got a composite of hydroxyapatite, collagen peptide, carbon dot composite. You can see from this electrochemical impedance spectroscopic data that actually these are the two reactants. Blue and green was blue and red was the two reactants, that is carbon dot and collagen peptide. Then these two forms first. It is a two-step process. First, it forms a composite, and it gives the last third and blue one is this. This one is last one is their formation, and then when it reacts with the hydroxyapatite, it is further decreased, but it behaves like a potential diagram. We can say that it is clearly formed this composites, and this your application we have done most of the test parameters. We can see that it is the FTIR spectra of these materials. Next, please. And what we have, we can say that fish waste is one fish waste, and we have to find out some more sources. And fish scale is one of the important sources for hydroxyapatite and beta dicalcium diphosphate. And then this hydroxyapatite derived fish scale is what is the advantage to the hydrothermal process is it is very nano-sized, and the crystal clarity is very white, pure white, and we have to this what we can say suggest that this along with hydroxyapatite and beta dicalcium phosphate together we can combine, and we can do some research as utilization as a bioceramic applications. Hydroxyapatite composite had potential application in pre-bond regeneration. It needs to be done. I think my friend Vinci has already told all those things, and now I will stop. Thank you. Next, please.
Thank you, Mohammad Ashraf sir. Like it was nice hearing to you, like about on hydroxyapatite. Hydroxyapatite being extensively used biomaterial for in bone, even in tissue engineering, dental application. Like this new, I mean, like this natural source, like it appears to be very appealing.
Yes, sir.
Hydroxyapatite source, source from fish scale.
Fish scale.
So it was a nice presentation. Like if somebody has some queries or questions, like forum is open.
I think not much questions. They are after lunch, no? Okay. And Cheryl Madhu has asked for raised his hand, please.
Yeah. Yeah. Sharon, madam, you can ask your question. Sharon, madam, can ask you your question.
She has to unmute, I think.
Ma'am, are you not able to unmute yourself? You can post a question in the chat box. So I presume there are no questions.
Yeah, it's okay.
Okay.
Okay, thank you, sir. Thank you for giving me the opportunity.
Yeah, thank you
very
very much.
to...
Yeah, thank you very much.
Okay.
Yeah, thank you very much. So I think we can move on to the next speaker. Next speaker is nobody but Dr. Santosh Pillai from he's professor and head Department of Pharmacology, Pushpagiri Medical College, Tiruvalla. And he will be speaking on the topic preclinical analysis of natural hydroxyapatite. I think he will be covering on efficacy of natural hydroxyapatite among the in vivo models. Over to you, sir.
Thank you, Sachin sir. Nice to see you.
Yeah.
So, hope my screen is visible.
Yes, sir, you're visible, sir. Your screen is visible, sir.
Thank you. So, thank you, Dr. Sachin, for that brief introduction. Good afternoon to all. Today, I'll be speaking on the preclinical analysis of natural hydroxyapatite, the in vitro analysis, and the natural hydroxyapatite. The chemical composition has been described in detail by the previous speaker. So, just to introduce preclinical testing of medical devices destined to be implanted in the human body must be first evaluated by a series of in vitro and in vivo tests before they are approved by the regulatory authorities. And what is the main aim, or what's the main goal of these preclinical testing? First, to determine the biocompatibility response when it is implanted into the host tissue, and the ability of a material to perform with an appropriate host response in a specific situation. That is, whatever clinical situation you tend to perform. And to analyze the potential for any unacceptable adverse biological effects when these biomaterials interact with the host tissue. So these are the main goals of doing preclinical testing. The good laboratory practice principles, FDA has developed its own guidance for preparing pre-market approval applications, and the evaluation for safety has been standardized over the years by the International Standard Organization. Now, the safety evaluation, the biomaterials are screened for their innocuity, safety, efficacy for their clinical indications. And biocompatibility test contributes to reducing the animal testing procedures by following the usual 3 ethical R's that we always follow during animal ethics, that is a replacement, reduction, and refinement. Now, hydroxyapatite biomaterials based on calcium phosphate, especially hydroxyapatite, are widely used to repair bone fractures and other defects, mainly because of their osteoinductive and osteoconductive properties. Osteoinductive property is a process by which there is stimulation of the osteogenesis, and the osteogenic cells are stimulated or shared near the organs or the tissues of injury. And osteoconductive is a process where the bone grows into the surface, that when it goes into the scaffold or implant. The other features that makes hydroxyapatite attractive for use as bone replacement is it is a major component of bone and heart tissues. It's non-toxic and non-immunogenic. That's why it is widely used in orthopedic and dental applications. Now, some differences between the natural and a synthetic hydroxyapatite. As Mohammad Ashraf sir has already described about the various natural materials such as the fish bones, fish scale, and mineral materials that has been used to produce a natural hydroxyapatite. We have synthesized hydroxyapatite is different from the natural one in terms of their strength and chemical composition. And in that aspect, the synthesized hydroxyapatite has got a lower biological activity than the natural hydroxyapatite. Now, this low biological activity can be overcome when it is preserved at a nanoscale level, and that will the beneficial effect could be achieved, and the biomaterial could be made more acceptable by using the nanoscale level of hydroxyapatite. Now, preclinical test where we undertake the biocompatibility and safety evaluation in vivo. We use different animal models, acute toxicity, acute systemic toxicity, where we explore the toxicity, systemic toxicity of hydroxyapatite. And this explores a systemic response in mice by the degradation products in aqueous and non-aqueous extracts of the sample as per ISO 10993. Healthy and active albino mice in the body weight range of 17 to 23 grams of either sex are used. And after acclimatization, the test and the control, we commonly use cottonseed oil, is injected intraperitoneally at the dose of 50 milligrams per kg. And each animal, each mice is observed at 4 hours, 24 hours, 48 and 72 hours for any evidence of abnormalities, with loss of body weight and death of the animal. Parameters that usually see are for respiration, motor control, convulsions, reflexes, cardiovascular signs, GIT abnormalities, piloerection, analgesia, and so on. There's a natural centrifuged hydroxyapatite, and on the right side we have the cottonseed oil that is used as control. And the test drug being given intraperitoneally into the mice and control given intraperitoneally. Pyrogenicity test, the test includes pyrogenicity of both microbial as well as microbial endotoxins as well as non-endotoxins, and we use mice after acclimatization is used for the study. The body temperature of the animals are recorded at regular intervals, and 3 animals are selected whose temperature is less than 39.8 degrees. The rectal temperature determined 30 minutes prior to the experiment is taken as the baseline value. The extract in the required dose is injected intraperitoneally, and the temperature recorded after 1 hour, repeated at 30 minutes interval up to about 3 hours. So we usually measure the body temperature, rectal temperature of the animal at graded intervals. Then we look for the irritation potential, mainly to see the local tissue response in the form of signs of inflammation, features of inflammation. So this is usually performed using a series of localized subcutaneous injections in order to preserve the biomaterial reactivity. And it is typically performed in rabbits utilizing multiple subcutaneous injection sites especially on the dorsum of the rabbits and on either sides of the spinal column, in order to determine if there is a presence of, if the presence of biomaterial extract causes any irritation or whether there are any signs of inflammation in the animal. And we use hydroxyapatite saline extracts are injected intracutaneously at 5 anterior sites at 2 centimeters apart on the left side of the animal, and saline controls are injected at 5 posterior sites on the left side. So you have the test as well as the control on anterior as well as the posterior sides of the animal. All sites are then evaluated for skin reactions like erythema and edema after 24 hours, 48 hours, and 72 hours following injection. And the difference in the sum of average scores for the control group and the test group corresponding to both erythema and edema indicate the irritation potential of the material. So whether this biomaterial is able to induce any erythema or inflammation in the host tissue. Skin sensitization assay. This is to evaluate the allergic potential of hydroxyapatite. It's divided into 3 phases. You have the induction phase 1, induction phase 2, and the challenge phase. And these phases are conducted in guinea pigs, which is considered to be a highly sensitive model for these particular type of studies. Okay. So initially we sensitize the animal with graded doses of hydroxyapatite. Then we put a patch over in case of the induction 2 for about 7 days and in challenge for about 14 days. And reactions such as erythema and swelling are scored at 24 and 48 hours following the removal of the patches. And we have a graded scoring system where 0 indicates there is no visible reactions, 1 indicates there is some discrete or slight erythema, 2 moderate or confident erythema, intense erythema or swelling is grade 3. So grades 1 or greater in the test group animals generally indicate sensitization. Then hemocompatibility. Hemocompatibility are used for devices that comes in direct contact with the circulating blood. Hemolysis, complement activation, and thrombogenicity testing may be performed. And hemolysis determines the degree of erythrocytes, red blood cell lysis, and the release of hemoglobin caused by medical devices in vitro. So this is usually done for those biomedical devices that comes in close contact with the blood, especially catheters and so on. Thrombogenicity. Safety testing for medical devices may also include thrombogenicity assessments, evaluation of biomaterial, associated thrombus formation, and subsequent thromboembolism to the downstream organs. It is performed in vivo using specific animal models, and then we evaluate the growth of that particular organ and also histopathological evaluations of these tissues. Alternatively, we do a series of in vitro tests for thrombogenicity that includes platelet adhesion and activation of the coagulation system, including TAT and partial thromboplastin time. Carcinogenicity. According to International Standard Organization, unless there is previously published information, testing the potential carcinogenicity of the implant may need to be explored. That means when we plan to undertake any carcinogenicity in the animals, we should have some published data that this particular biomaterial does not have any chemical that can cause harm to the animal. So the aim is to determine the tumorogenic potential of medical devices, materials, and other extracts. So that's what ISO guidelines have been specified. So unless we have any published data, we cannot undertake any potential carcinogenicity test in animals. Long-term studies in rodents for about 1 to 2 years, though transgenic models have also been recommended in recent years and may be considered. Coming to implantation, central component of biocompatibility test is the biomaterial's local effects after implantation. When it is directly implanted into the animal, how does it react in the body of that particular animal, how it interacts with the surrounding tissue. And these are evaluated by comparing the biological response of the test material against the control that is recognized for its known biocompatibility and inertness. The objective of this particular test is to evaluate the tissue response elicited via the biomaterial following its implantation, integration, and biodegradation, how it behaves in the host tissue. So subcutaneous implantation pockets are created, and then the biomaterial is being implanted into the animal. Histopathological analysis is being done on HNE, and in the test we can see the calcification marks and the giant cell response here. The control also we can see cross calcification. So this being taken into the host tissue without any sorts of inflammation. Then there's a long array of list of performance and efficacy evaluation. I'll not be going into much details. These tests are mainly to understand the performance of hydroxyapatite in the actual clinical settings. So various animal models have been developed over the years in order to closely match the clinical conditions. So the ISO guidance suggests to perform a comparison between the test and the control at the similar location. So we have the test as well as the control at similar locations at different implantation sites. And along with that, we can also study the systemic effects of the biomaterial. The same animal model, we can also do a second study to understand the systemic effects of the biomaterial that includes the daily or weekly health evaluations, body weight measurements, histopathology of the biomaterial and downstream organs, and clinical pathology at termination. So we have a long array of tests that we can do actually on the specific bones that we intend to study in the clinical settings. So just to conclude, safety and efficacy models for orthopedic biomaterials is not exhaustive, as this is a continuously evolving field, and the evaluation of biocompatibility, safety and efficacy is of primary importance in determining the best materials for treating various orthopedic dental conditions. Future directions will be dependent on the newly developed products, and antimicrobial combinations, products, and stem cell research could play a bigger role in biomaterials in the near future. Thank you.
Thank you very much, Dr. Santosh. In fact, you have given what you call an exhaustive overview about how a material, any material can qualify as a biomaterial. So you have covered like safety evaluation, then performance evaluation, efficacy evaluation. In fact, like you have covered almost like everything. Thank you. Now, forum is open for discussion and questions.
Sir, can you hear me?
Yeah, Marin. Yes, please. Yes, please.
Sir, what is the difference between doing this procedure in the ISO certified lab and the conventional normal labs? So how it should be initially, how you should proceed with. So sometimes if you go to the clinical trial, they may ask that it has to be done in the ISO lab, and otherwise we'll be doing in the basic laboratories. So how you should select a procedure, how to proceed with once you develop a material like this. So how we should proceed with it? Directly we should go for a ISO lab, or we should do the initial studies and then you should proceed to the ISO labs.
I think Dr. Sachin will be able to explain more on this. I think basically we can have these tests done in our routine labs, but mainly for documentation purpose, especially when we are going for clinical trials. We ask for this ISO specifications. So I hope it's right, Dr. Sachin.
Yeah, in fact, like when you do these tests in an accredited platform, like the data is more acceptable to the regulatory authorities. Otherwise, you need to justify, like if it is done in an accredited lab, like that makes things easier. Like to get approval. So,
Okay, sir. Thank you.
okay. Yeah.
Yeah, okay. Actually, thank you for giving the excellent presentation. I want to know that which part of the fish bone you have used, like natural hydroxyapatite you have used for clinical validation. So, it means that what is the size of the nanoparticle or
We use a fish scale that is naturally available. Fish scale.
scale? Okay, okay, right.
And it's being tested along with the control where we use cottonseed oil as a control.
Okay, right.
Sachin, sir, you should unmute, I think.
Sorry, sir. So if there are no more questions, shall we move on to next speaker? Yeah, somebody has raised. No, somebody has raised the hand. Yeah, Meka.
Yes, sir. Let's move to the next session. Kindly moderate.
Yeah. So our next speaker is Dr. J. Venkatesan. So he's assistant professor, Yennepoy Research Center, Yennepoya, Mangaluru, Karnataka. And also he is associated with Hamgyong University, South Korea. He will be speaking on biological and biomedical applications of naturally derived hydroxyapatite. I think he will be covering various areas of utilization of hydroxyapatite in general medical field. Over to Dr. Venkatesan, sir.
Thank you so much for your kind introduction. Let me share my slide.
Sure.
Oh, is it visible, my slide?
Yes, sir. Yes, sir. It is visible.
Yeah, this is one of the word I mostly used in last 2 years. Is it visible, my slide? Because whenever I give lecture on, most of the time I give in lecture in online platform. Okay, thank you so much for giving this great opportunity to present my work here. I'm Dr. J. Venkatesan. I'm working as an assistant professor in Yennepoya Research Center, which comes under Yennepoya Team to be University, and it's located in Karnataka, Mangalore. And also I'm the visiting fellow in Anyang University, located in Seoul and South Korea. So actually in last 2 talks, we have extensively seen on hydroxyapatite utilization and isolation, as well as some of the ISOs, how to make the clinical validation of hydroxyapatites. Actually, I'm a basically chemistry related person, and we work on biocomposite for tissue engineering applications. So these studies I yearly done in my PhD related work, and the topic of my presentation, biological and biomedical application of natural derived hydroxyapatite. So today I will be giving some brief information on history of biomaterial and what is tissue engineering, and that is the main theme of the current conference. And hydroxyapatite and the role of hydroxyapatite in bone tissue engineering, especially in orthopedic application in long bone fracture, and utilization of hydroxyapatite in dental application, and some of the brief information about the commercial application and the future approach for the hydroxyapatite, whatever we have used for our experiment. And history of biomaterial. Actually, biomaterial have very long history. We have to go at least 7,000 years before to know about what biomaterial means. So in ancient time, you might have seen the war usually go on between the 2 kings, and the many soldiers got affected. They lose their limb or finger or even any part of the body. So usually they cover with a leather or wooden material, and to make like a cosmetic is similar to the organs, but the main function that is no function is the usually it may more or like a cosmetical arrangement. But after developing the industrial revolution, especially for um, coming into like metal based materials, and you can see the Peter Stewart in 1600, so they used metal implant. It's not only working as a cosmetic leg like material, and also it's functioning as a load bearing material. So we all know that in the top of the picture, the bridge in tooth repair, it's working in so long time. Like in this case, the gold and the natural teeth, they have used like a bridge material. So after come to the developing the polymer material, then there is a significant changes, the improvement in biological derived material, especially for the polymers, like a natural polymer as well as the synthetic polymer. Some of them natural synthetic polymer, they already commercialized. So one of the best example is the hip replacement. In most of the time, the clinician used metal based rod, like it made up of titanium, and these days they are replacing it, or they're replacing one part of the material using PEEK or other kind of polymers. And recently what we are going to do it like artificial skin and 3D printed organs. These are all the very emerging area of research, and which can be used for in biomaterial as well as for replacing the organ. It's not only using the cosmetical or mechanical, and is integrating with our body. Is like, for example, in ear or even the finger or even the nose. So it has the enormous application. Application is there. And again, one more interesting point I want to tell you, in Robert Vacanti in MIT in 1997, they have developed one ear mouse. So actually that one is not coming up, but there will be something like a gene editing or CRISPR things. So still they are working on that particular area. But when you go into the artificial developed organ, so it really need one multidisciplinary field of research. So starting from the material scientist and bioengineer, even chemist and biologist and computational scientist and immunologist, even the surgeons. For example, the problem or the challenges what we are facing in biomaterial. So one group of people, they are working on particular area. For example, the material scientists, they develop one composite material. After that, they may be published or even they patented it. And after that, they are not taking over into the next step because their laboratory may not have sufficient instrument or other challenges. So the next time the other person, the biologist, they do some kind of in vitro study, like do some gene expression or some other kind of related experiments. So still it's not going to end. Again, they have to go to the surgeon and they have to do some clinical validation, as Dr. Santosh Pillai explained very detail about the histocompatibility, sensitivity, thrombogenicity, carcinogenicity. So we have to follow one long procedure of ISOs. Then we can take that material into the clinical level, and DCGI, they have to approve it to take that material into the commercial levels. So those things I'm going to tell about in last of my slides. And what is the history of biomaterial? This much. And so what I'm going to talk about today is mostly based on the bone. Bone looks like a very simple structure, but the arrangement of hierarchical arrangement is really making very complex structures. So when you come to the left side from the right side, left side to the right side, it's made up of the collagen moieties as well as the collagen moieties. Again, the hydroxyapatite crystals are arranged in the uniform manner, and it's making like some kind of bundle structures. So that bundle structure as well as in the nanofiber form, it's around osteons or even the blood vessels is going through. This kind of arrangement, we have to try to make it using any other methodology, whether 3D printing as well as like any particle leaching, or even we can integrate it with the using stem cell technology. So why we need to make the bone? Because there are a lot of incident is going on, starting from the motor accident or bone fracture or osteonecrosis or even birth abnormalities or even tumor resections and several other problems like osteoporosis or arthritis, which leads to the bone defects. We need to make some kind of treatment options. So are there any treatment option in our current medical field? Of course, we do have the three important treatment options. One is autograft and allograft and synthetic grafts. So you all guys know very well about this. What is autograft? Autograft is nothing but taking the same person bone in from the iliac crest, and then they can replace it in the defect area. And allograft is most of the time we use the cadaver bone and stored in the bone bank. Whenever they need it, they can take it out. And again, the another portion is a synthetic graft. It's one of the area is just booming up. So what is the problem with autograft? The main issue with the autografts are insufficient donor site and need secondary surgery. And the problem with allograft is non-osteoinductive, and sometimes the disease can be transmissible from the old person to the new persons. But the overall, the treatment option cover around more than 90% the clinician do with these two grafts only, because the success rate in autograft is really very high. So they use for the synthetic graft, it's made up of like ceramics and even hydroxyapatite based material they used currently, like a bioas from the natural source and pepgin and bioactive glasses 45S5 and bioactive glass composites. So this material is already commercially available based on this hydroxyapatite, but still there are some issues, especially for non-osteoinductive. This is one of the biggest problem in whatever the synthetic material we do have it, because they can be osteoconductive but may not be osteoinductive. And sometimes when you are placing this material, sometimes it's the leaching can takes place in the, and also the bioas and all is really very expensive. Even the milligram is go to some rupees in thousands. And what are the biomaterial available for tissue engineering? We can classify it broadly like a polymer, metals, ceramics, or carbon based material. And again, we can separate it as a natural biomaterial as well as for synthetic biomaterial. The natural biomaterial, again, we can separate it to the protein-based one and the polysaccharide-based one. The protein, silk, collagen, or fibrin, or gelatin, most of the time they take it from the fish source or pig or even ox-based origin. And the polysaccharide, hyaluronic acid, alginate, agarose, and chitosans. And coming into the synthetic side, a PEG, polyethylene glycol, polyglycolide, and polylactic co-glycolic acid, PLGA, and PDLLA, and then PCl. So this comes under polymeric category. And ceramic category, it's again like a tetracalcium phosphate, hydroxyapatite, bioactive glass, and calcium phosphate, and alumina and zirconia. There are some other kind of material like a carbon-based material and even the metal-based materials. So this metal-based material like titanium or zirconium, magnesium, even some kind of transition metal dicholgogenides, it's still going on. So these all material they are trying to utilize in tissue engineering for various purposes, especially for the bone, they use it. So coming to what is tissue engineering, what's the basic paradigm for tissue engineering, what is exposed in around 20 to 30 years before they started it. And the tissue engineering is a subfield of biomaterial, is truly interdisciplinary research, as I told you in my first slide. The main function, but the role of tissue engineering is to develop any artificial organ or tissue which can replace or maintain or improve the tissue function in the given the whole area or in the native tissue. So when we start, for example, in my case, we are developing bone tissue. The first one is first we take some kind of biopsy. Most of the time we take some kind of bone marrow or bone biopsy. Then we isolate some kind of osteoblastic cells, isolation of the cells. And then we multiply with the tissue culture plate in million level. And then again, we multiply into the billion level. And we made the scaffolds. So this is the very important step, the tissue development, which made up of the scaffold, and we add the cells, and sometimes we add nanoparticles as well as some kind of growth factor or even the mechanical stimuli and extra and etc. So this case we call it bioreactor system. So once we develop any tissue and we can implant it back, it can work well. So this is what the basic paradigm they are talking about, but still we are going on this method only. So here we can see that the scaffold play important role, how we can make the scaffolds. So we all know that the clay, we everyone know very well about the clay. Even in the childhood, even we could have seen that they make some kind of process. The clay, they can convert it into the pot. So the same thing here also, we have the polymer, ceramic, metal, and carbon-based materials. So we need to know some kind of process. So this process can be in any form, that is the electrospun fiber or gas formed technique or the salt leaching method or free fabrication method or even the 3D printing or 3D bioprinting or additive printing. There are several kind of metal. The thing is what structure you need it. For example, in the clay, we need to have some kind of pot-like structures. So in using the any other methods, you can create a pore-like structure or highly mechanical strength, compressive strength having like more than 100 MPa structure material we can develop using any kind of technique. It varies from one tissue to the another tissues. And coming to the synthetic hydroxyapatite. So synthetic hydroxyapatite is one of the well-known method. Even they started around 1960 to 1970. They developed it. Synthetic hydroxyapatite is one of the well-known method. Calcium nitrate and ammonium phosphate and adjusting with the pH. Like a calcium precursor and phosphorus precursors, they use some kind of water as a solvent, and then they immediately form it. It's formed like a milky product, immediately can form it, and then adjusting the pH around nearly 10, and we have to keep for the aging with the low temperature, and the crystal will form. And we have to keep for gelation for time for some more time, and then filter it, and then calcined at around 800 to 1000 degrees Celsius, we can get it hydroxyapatite. It's one of the very simplest method and well straightforward method, and of course it's a very inexpensive method. And what is the problem with this method? Why we want to go with the natural hydroxyapatite? So there are some problem with this method. The problem with this method is this method is very sensitive, especially when you are going with aging and gelation. Most of the time, instead of forming hydroxyapatite, there are some trace level of some other bibasic material like octacalcium phosphate or beta calcium phosphate or even some other kind of phosphate. It's formed together. The calcium phosphorus ratio is less than 1.67. So that time we may not use it as a bone graft substitute. So that's why we go for natural sources for hydroxyapatite. In natural sources, of course, all the bone, even the human bone, also can be utilized for isolation of hydroxyapatite. The commonly used natural source are bovine bone and chicken bone and marine sources, especially fish, scale or skin, scale and bone, and even some of the egg part they can utilize it, and the eggshells and muscle cells and the snail cells and some of the marine mineral resources like a calcium based resources, and then they can use it some different kind of method and they isolate the hydroxyapatite. So in hydroxyapatite can be isolated in multiple way. One is the thermal calcination. Is one of the well-known method. Just directly take the bone and then put it into the muffle furnace around 900 degrees Celsius or 1000 degrees Celsius for 5 hours. And of course, the collagen and other moieties, definitely it will remove it because of that high temperature, and then finally we get hydroxyapatite. And another one is alkaline hydrolysis method using the sodium hydroxide. Is one of the commonly used chemicals to isolate hydroxyapatite, and some combination of hydrothermal method and some other like a salt gel and the mechanochemical. These are all some of the method they used it, like microwave and then pyrolysis and some other case. So what we are doing so I would like to present what we have done it, like fish bone from we call it waste to the best, and we make some kind of scaffold things. And the isolation of hydroxyapatite from fish bone, we follow the two different methods. One is thermal calcination method and another one is alkaline hydrolysis method. The first, we collected the fish bones from the local market in Mangalore, and then we crushed it, and then we just used thermal calcination, just placed in a muffle furnace, and then we got it within 5 hours. We got hydroxyapatite. And another one is alkaline hydrolysis method we used, and we got the pale yellow color hydroxyapatite. And then we characterized it. Actually, one more very important difference between natural hydroxyapatite and synthetic hydroxyapatite. We can see that in the right side FTIR column, and there is some peaks are missing in 1600 or around 1500 in this sigma derived hydroxyapatite. The D is sigma hydroxyapatite, that means the synthetic hydroxyapatite. But in the case of C and B, and we can see that some of the peaks like 1455, 1417, these are the carbonated hydroxyapatites. These carbonated groups are not present in the synthetic hydroxyapatite. These carbonate groups in the literature, they said it can be induced the bone formation much faster than the natural derived hydroxyapatite. So coming to the left side, the thermal gravimetry analysis. So this method, even the alkaline hydrolysis method, even thermal calcination method, is completely removed the collagen organic moieties. That means 365.6 is related to collagen moieties. And coming to the purity of this material, and the purity, it's really very high pure, because the thermal calcination method, we can see that the thermal calcination method is more like a white, and the crystal size is around one micrometer size, whereas in the alkaline hydrolysis method is produced the nano size hydroxyapatites around 17 to 70 nanometer. And we can see that the fish bone is really, the fish bone, the what we have used is really quite big, and we can see that it's a bone like structure. The collagen and other organic the crystals are placed in the matrix. And coming to the XOD data, we can see that B and C. The B is alkaline hydrolysis method, is somewhat is amorphous in nature, but the still retained is peaks around 31 degrees Celsius, whereas on C column is thermally derived hydroxyapatite. We can see almost equal to the synthetic derived hydroxyapatite, which is present in the D form. So we can say that the fishbone derived hydroxyapatite is pure as the synthetic derived hydroxyapatite in terms of chemical composition. And then we can see that here the particle size, that TEM analysis indicates that the alkaline hydrolysis derived hydroxyapatite shows 17 to 71 nanometer, and EDX spectrum is equivalent to the XOD data. And alkaline hydrolysis method is, you can see that it's around 1 micron in size, and the crystals are even well crystalline in nature. And the calcium phosphorus ratio is around 1.67, as we indicated in my previous slides. And what is the difference? Actually, we compare with JCPDS data 090432 and the thermal calcination method, and even the alkaline hydrolysis method, and a different kind of crystallographic plane, HKL plane we presented here. And we see the total error, and we calculated between the JCPDS data and obtained hydroxyapatite using the two different methods. One is thermal calcination and another one is alkaline hydrolysis method. The error is really very less, which indicates that the derived method is almost equal to the JCPDS derived hydroxyapatite. And then we check the cell viability with using MTT assay. The derived compounds are not showing any toxicity. It's biocompatible. And also we do some experiment with OH staining. And then once we got hydroxyapatite, of course, the hydroxyapatite alone cannot be utilized for bone tissue engineering. It has to mixed with some other component. Most of the time, the polymer and ceramic based composite, they extensively used for tissue engineering applications. So then we used for well-known material, that is hydroxyapatite. So here, what we have made it, we take the fish and then we get the fish bone, and then using two different methods, alkaline hydrolysis as well as the thermal calcination. And thermal calcination produce micro size hydroxyapatite, and alkaline hydrolysis produce nano size hydroxyapatite. And we got chitosan from the crab, and then we mixed with chitosan and hydroxyapatite, and we made it two different kind of composite material. One is chitosan micro hydroxyapatite, and another one is chitosan nano hydroxyapatite. And we observed the surface morphology using scanning electron microscopy, which inferred that the scaffolds are really highly porous in nature, the size around 100 to 500 micrometer in size. And then we added again some other kind of material, like improve the mechanical strength, the carbon nanotubes. And we can see that the chitosan have the improper pore structures. By adding hydroxyapatite in the matrix, it produced some uniform and as well as the interconnected porosity, and the dispersion of hydroxyapatite uniform in the matrix. And also we checked whether the scaffold is integrating with osteoblast cells-like cells, and we can see that here the scaffolds and the cells are merging together and they are growing it more reliable way, and which indicates that the developed scaffolds are non-toxic in nature. And then we checked with mesenchymal stem cell using adipocyte-derived mesenchymal stem cells, and we checked with the cell proliferation in different days, 1 day and 3 days and 5 days and even the 7 days, and with both the scaffold chitosan micro and the chitosan with the nano hydroxyapatite. The cells are growing with the period of time with increasing. That means the scaffold doesn't show any toxicity. That means the scaffolds are biocompatible. Then we implanted this scaffold in the rabbit femur, and the rabbit femur we cut it in the small 5 centimeter, and we placed this material, and then we checked the radiography at radiography for the 8 weeks, and we can see that the group A and the group B and the one control group, of course, we just compare with the chitosan micro hydroxyapatite and chitosan with the nano hydroxyapatite. Then we observed that the group B is shown better result than compared to the micro hydroxyapatite, which evaluated with micro CT. And then we checked with histological staining and the other parameters like bone volume and bone tubercle volume. And the group B compared to the group A, and we observed that a group B shows better results in terms of bone forming ability. As a conclusion of this work, like hydroxyapatite from the marine fish bone, one of the best ways, the inexpensive, and can be potentially useful for the addition of other polymeric substances. Of course, the chitosan is one of the best material we can combine with, because it can be used as a organic material, and other material like chitosan, hydroxyapatite, even the carbon nanotubes to increase the mechanical strength, as well as some other gene expression, alkaline phosphatase and mineralization effect. So we need to have some kind of tri-component system to achieve the goal of to mimic the natural bone, and total volume, bone volume, and other important parameters are increasing while adding nanostructure hydroxyapatite when we compare to the microstructure hydroxyapatites. So the nanostructure hydroxyapatite having the potential application in bone tissue engineering. So so the another part is you might have seen that cuttlefish bone. The cuttlefish bone is another kind of bone. The important difference between the normal bone, normal fish bone, and the cuttlefish bones are the normal fish bones is like a needle-like structures. Usually, it's like a branch structures. But in the case of cuttlefish bone, it has a surface morphology. It has the dorsal shield and the lamel, and then it has some highly porous structures. We can see that the figure D, it has a very good pore structures. And we can cut it into as a bone graft directly. We can take the bone, and it's really a very sponge-like substances, and we can cut it as a bone graft substitute. And one more important difference between the normal fish bone as well as the cuttlefish bone, the chemical composition. The chemical composition is actually in normal fish bone is hydroxyapatite, but in the case of it's more or like a calcite or other calcium-based material. And you can see that the thermal gravimetry analysis, even you can see one strong deflection around 600 degrees Celsius or even the 700 degrees Celsius, which means that around 50% composition is disintegrating, which means that even the hydroxyapatite is very stable, even 1200 degrees Celsius. But in the case of cuttlefish bone material, it's not hydroxyapatite. So we need to do some kind of process. Even the XRD results suggest that there is no peak around 31 degrees Celsius. So there are so many other peaks, which may be this other form of calcium phosphate. So what we have done we made it the cuttlefish bone, and then we just milled it, and then we add some kind of phosphate sources. And then once we add some kind of phosphate sources, and then we add like hydroxyapatite nanorod, we can get it. Like we can see that around 31 degrees Celsius, we can get hydroxyapatite. So what we have to do it here, we need to know some kind of sulfate precursor. We need to make hydroxyapatite from the cuttlefish bone. So the cuttlefish bone can be changed into different morphological structures. Like we can go with square and even some rectangle side, or even some roll side, or so the same any structure we can make it out. Even the clinician, they can directly shape it out using that trimmer, and then they can implant it, and then they implant it into the person, and then they just check the histology and radiological analysis. So it's giving the better results from the cuttlefish bone hydroxyapatite. So the cuttlefish bone hydroxyapatite is one more alternative source for hydroxyapatite, for which can be used for tissue engineering application, and we can use the combination of some other polymeric substances like a polyvinyl alcohol or PCl, or even some of the other material like collagen gelatin or even alginate to increase many other functions. And incorporation of the stem cells, of course, it can be, we can do that, as Dr. Santosh Pillai said, that the stem cell is one of the burning area of research in tissue engineering, but still we have to go very long way. Of course, the long animal model, we need it like monkey and dogs to utilize the cuttlefish bone hydroxyapatite with the stem cells. So come to dental application. So can be utilized for hydroxyapatite from the fish bone for dental application. The dental application, there are two or multiple application, like one is the bone graft as well as for remineralization potential of the decay teeth. So dental decay is one of the very common problem. Almost around 90% of the Indian population affected with the dental caries. At least they can visit one time of their lifetime to the clinician. So usually they treat with multiple treatment. So one is the nanoparticles. Still these are in the very earlier stage of research, especially the silver nanoparticles, nanoparticle based on silver. And then they can treat it with the remove the infection first, and then they can use some kind of other material that's we call it regeneration materials. So currently the market is really huge on dental based dental caries. So people use for composite amalgam or glass ionomers or even the gold filling. So what we have done it, so initially we developed silver nanoparticle, and then using chito-oligosaccharide is one kind of polymeric substance, is the derivative of chitosan. The chito-oligosaccharides are very small molecule, and it is easily soluble in water. So what we have done it, the first we take the silver nitrate and chito-oligosaccharide, and then we just add the microwave method. Actually in microwave we directly add with chito-oligosaccharide and the silver nitrate precursor, and then within 10 minutes it's formed the silver nanoparticle. The yellow color indicates that the formation of silver nanoparticles. We can see that around 414 nanometer we can see the silver nanoparticles formation, and then the silver nanoparticle size is around 32 nanometers, and FTIR and XRD results suggested that the purity of derived silver nanoparticles. And then we done with FESM and transmission electron microscopy. The transmission electron microscopy, you can see that the dispersion of the nanoparticles in the carbon grids are uniform, and almost the particle diameter is around 14 nanometer in size, and also it's more like a monodisperse in nature. And then we made some kind of filling material with a combination of silicone oil and adding with calcium hydroxide and cos and silver nanoparticles and ceramic powders, and we made like a paste kind of material which is insoluble in water. We can use as a dental pillar. So, and also whether we can use for natural derived hydroxyapatite from the fish bone, whether for the remineralization potential or not. Then we have taken with the premolar teeth and 51 teeth, and then we do some experiments, and we have taken the group 1 and group 2 and group 3. The first group is a control one, and the second one is a micro hydroxyapatite, and the third one is nano hydroxyapatite. And then we made artificially made some kind of caries, and the right side same images you can see that the figure A indicates the caries same image, and then the C, and then the group B, group 3, we can see that the D is a group 3, and the group 3 is nano hydroxyapatite, and the group 1 is commercial product, that means the control, and then the group 2 is micro hydroxyapatite. And we can see that the hydroxyapatite from the fish bone, it has the remineralization potential. So that means that we can utilize hydroxyapatite from the fish bone for remineralization of the dental teeth, along with the nanoparticle addition, like even the silver nanoparticles. So again, we made some kind of other kind of material in a microsphere. One is calcium chitosan and chitooligosaccharide silver nanoparticle with the combination of hydroxyapatite. And this method is very simple and straightforward. The first, we have taken chitooligosaccharide silver nanoparticle and add some kind of alginate into that. We make some kind of hydrogels, and then we add hydroxyapatite to form some uniform slurry-like material, and then we dropped it into calcium chloride around 10%, and it forms the microspheres. And this microsphere is really good in size. The size is around 1 micrometer in size, and this material along with silver nanoparticles, we can see that the first one is without hydroxyapatite, but when addition of the different concentration 1 mg and 2 mg and 3 mg hydroxyapatite, we can see that the particle size keep on increasing it, and we need to give some kind of optimization. So finally we observed that chitooligosaccharide silver alginate with hydroxyapatite and 1 gram hydroxyapatites give the better shapes as well as the better biological activity. And then we check the antimicrobial property of the developed material, and then all the three material shows a better inhibition and Streptococcus aureus, and of course is the addition of the silver definitely give better results. So what is the conclusion of this portion? Chitooligosaccharide silver nanoparticle addition with the combination of polymer and hydroxyapatite from the fish bone can be utilized for dental application as well as for bone tissue engineering applications. So other kind of polymer like alginate or even the silver play essential role is not only giving the matrix to the scaffold structure and also give some kind of microbial inhibitions, and antimicrobial properties essential for especially for the long-term bone graft materials, especially for the implants. So this silver nanoparticle can be utilized. Of course, we need to go very long way. We are in very early stage of the research, and even we done with some animal models. Still, we have to go some ISO procedure to check the hemocompatibility, sensitivity, and thrombogenicity, and carcinogenicity. Even we have to implant in the higher animal model, like as I suggested, sheep or dog, and to know the exact function of this material, then we can take it in the commercial levels. So this is what my presentation. I just want to show you some of my research center. Like we do have we don't want it. Okay. So we belongs to Empire Research Center, and we do almost more than around 24 faculty in our department, and it has some different kind of field of people like biotechnology and even nanotechnology, and around 70 JRF and SRF they are working on in different multidisciplinary field like microbiology and molecular biology. And we do have some cancer team, they especially working on cancer, and we people working on some proteomics and genomics for various type of cancer identification, as well as they're finding out the markers. And we do have some kind of bioinformatics labs, so they are working on especially the genomic and proteomics people and developing some kind of software. And we have some kind of stimulation centers. Maybe you can visit us anytime, so I will be ready for that one. So go for the next slide. And research and development and regulatory. So these things we have to think about commercialization, whether can it take it hydroxyapatite from the fish bone can for the commercial product. So this is what I aimed for several years before, but it has a lot of challenging, especially for getting the raw material. Getting the raw material, for example, the fish species can vary from its composition from one time to the another point of time. That is one greater challenge I have faced, because the uniform raw material getting for hydroxyapatite isolation is another challenging task. So especially the research itself takes 1 to 5 years, and the establishment of lab, and we have to go in vitro cell culture and setting up the animal model. It's really time consuming process, usually takes 1 to 5 years, and developing the scaffolding system and go to the clinical travel and follow all the ISO method, especially following the regulatory things. It takes another 3 to 5 years, and after that we can go to the commercial products. So what is the new technology we are going on? One is the electrospinning, and another one is 3D bioprinted 3D bioprinting even the any tissues like even the bone, skin, and even the liver, kidney, and go on with the heart or even the brain. So these are the very advanced technique which we need to adopt with whatever developed we material like etoxapatite can develop in the biomimicking property with the natural bone. So where are we in tissue engineering, especially for bone tissue engineering? As I told you, the calcium phosphate material, they started very early stage around 20th century, around 1940 or 1950. Even in the World War, they have used some kind of calcium-based material. And then people used bioactive glasses 4,5-S5 in 1960 to 1970. And then BMP, they developed this, one of the very important discovery in bone related research, especially for bone morphogenetic protein 2. And then they got the FDA approval. And then after in 1990, that is the very initial stage, they made the polymer and polymer matrix composites. Still we are following the same thing, and the addition of RGD peptides is also kind of replacement of BMP2, because the BMP2 is really very expensive material. So, but that is one of the very best material I can say, because it's inducing the osteoinductive effect in even the stem cell, because that is one of the essential parameter which I have to consider while doing for scaffold fabrication for bone tissue engineering. After 2000, even the 2010, they used for 3D bioprinting. So this is one of the hot area of research, 3D bioprinting, especially with etoxapatite or even any other material like PEEK or PLG or PCL. With a combination of protein or polymer or any kind of material, we can go for developing bone tissue materials. And where are we in tissue engineering? Of course, we are in most of the companies, they have developed epibone or even bone-based material. And the tissue engineered material, it's very limited, but still we are still depending on etoxapatite. Even the bioas product from bioas is one of the commercially well-known material, but still we are a very initial stage of developing any full functional organ, because the organ have the multiple stage. For example, the skin and bone, these are very early, means it do not have very complex structure as compared to the any other functional tissues like heart and kidney or even brain. So we are very initial stage. It takes some more time to develop this kind of full functional organ in tissue engineering. So I would like to acknowledge some of the very people, they helped me throughout my life, even in career. So the first one is Professor Sikwan Kim. Is my advisor in my PhD, and he's one of the top scientists in always by Web of Science, like Thomson and Tutor, and top one scientist. And also he is nominated for Nobel Prize. And he only taught me most of the thing, the fishbone isolation and all other experiment. And the second one is Professor Anil Sugumaran. Most of the dental work he taught me, and he gave me the idea. Is basically the clinician having the lot of research knowledge, as well as he has published a lot of papers on dental as well as the bone related area. He's working in Ahmad Medical College in Qatar. And my director currently, Professor Dr. Rekha, and working as a director in Enapa Universities. He's the one key person in my center, developing several technology. And she supported me in all the way to develop composite biomaterial for tissue engineering. And Professor Shambhat sir, she is the one of the very keen to help to anyone, and with as well as knowledge, as well as he give a lot of problem whatever he face it. And he work as a vice principal of a dental college in Enapa University. And all other my students who work as a JRF as well as SRF, and they most of the time they have done all experiment. Even some of the MDH student, Dr. Ashwati and Dr. Gopika and Dr. Sam Nipunika, they also working on some of my project apart from this etoxapatite. And this is my current team in YRC. So as I mentioned, we have some faculties. And thank you so much for your kind support and listen my words. And if you have any question or queries, please write me, or even you can message me through this number. Thank you so much.
Yeah, thank you, Dr. Venkatesan.
Yes.
In fact, it's almost like clear that you belong to a dedicated team of researchers, like who are extensively working on this biomaterials, especially for dental and orthopedic applications.
Yes.
In fact, you have covered multiple number of biomaterials like fishbone derived HA, nano, micro, composites with chitosan, yeah, and even like I think silver nanoparticle you were mentioning. So really, what you call extensive kind of research. Thank you so much for such a good talk. Yeah.
Right.
And now the forum is open for discussion.
So I got a question. What about the degradation rate of the natural hydroxyapatite? Usually there is an issue with the degradation rate of the hydroxide, especially the synthetic one. So it has got any added benefit if you take the natural hydroxyapatite?
Yeah. Actually, yeah, right. Actually, the adding advantage with the natural hydroxyapatite, the one is the carbonated groups actually that is present in natural hydroxyapatite. That one, the literature says that it induced the bone formation much more quickly than compared to synthetic hydroxyapatite. But in terms of composition, it's not much difference actually. But actually, the synthetic hydroxyapatite, the problem with that one is sometimes is brittle and also is leaching out from implanted sites, but that's why we have made most of the time composite biomaterials, so it can the polymeric matrix can hold it, whether that is synthetic derived hydroxyapatite or natural derived hydroxyapatite, it will be same more or like in terms of degradation. But we feel that the natural hydroxyapatite better source for bone regeneration, especially the carbonated group. That one point I can say that.
Okay, thank you, sir.
Right.
Any more queries from the participants, audience? So, since there are no more questions, like thank you once again, Dr. Venkatesha.
Yeah, thank you so much. Yes.
Thank you for.
Thank you.
Okay, thank you, dear Dr. Sachin, for the wonderful moderation of the sessions. And I would like to thank the speakers also, Dr. Muhammad Ashraf, Dr. Santosh Pillai, and Dr. J. Venkateshan for their fruitful sessions. Now, let's move on to the rest of the program for today. That is the hands-on training. So I would like to invite Dr. Shirley Anthony, who is moderating the sessions.
Thank you.
Thank you, Dr. Meka. So going on to the next program, which we have as a hands-on training, and the faculty for this is Dr. R. Ramya. She is a professor and head Department of Oral Biology, Savita Dental College, Chennai. So the topic ma'am will be taking is how to write a successful grant proposal, which is one of the basics for getting through the eyes of the different funding agencies. So over to you, ma'am.
Thank you, Shirley. Thank you. Is my PowerPoint visible?
Yes, ma'am, but can you just enlarge? Okay, yes, ma'am, it is visible.
Okay. Yeah. Can you see the next slide?
Yeah. Yes, ma'am.
Thank you so much. Yeah. So good afternoon. And I think you've heard a lot of deliberations throughout this wonderfully organized workshop and conference. And the topic especially is something which definitely is the need of the hour, as already said yesterday morning in my first lecture. And the vision behind such topics, the lineup of the topics were very impressive. Kudos to the organizing team and the organizing secretary, Dr. Nebo, and the brain behind, Father Matthew, sir. So here I am to give you a hands-on. Hands-on is not that easy to do it online. So I would just ask everyone to quickly grab a piece of paper. So you can just get the best experience of the next few minutes, which I'll be enlightening you about how to go about writing a grant proposal. So why was I chosen for doing this particular topic is because of my ongoing grant with BIRAC, and I have had very good experience with Department of Science and Technology, which actually helped me fetch All India Best Innovation Awards. And again, you know what research means, and all of you are researchers. But without touching these beautiful statements, we can't understand what the funds actually mean to highlight and understand or underline the importance of research. So whatever it is, the most important thing to run the research for the benefit of the larger society or your fellow beings, you definitely need funds for research. Without funds, it is very, very difficult to make your dream come true, make your ideas come true, or make your ideas be translated. So that is why we are here today in this particular workshop. And of course, funds are very, very important backbone of the research and development, which is vital. So are you all ready? So the basic requirements of research funding is definitely the first thing what you need to write is what is your expertise? Which area are you good in doing? If you are a youngster or you're a beginner, you've just joined in any institution or you've just begin your career in research, you can definitely tap in some grants which doesn't need any research expertise. Otherwise, 90% of the research agencies believe you only if the profile or your experience is good enough for them to believe in you to give that kind of money. Okay, so that means what are the publications which you have right now in your kitty, which are in high impact, or which are the major core areas of the publications you have is the most important thing which you have to remember. So take always those areas to win a grant. That is the key. So, and in case if you're just a beginner, so what do you do? If you're just a beginner, you can definitely think in terms of the particular area which really makes you very happy about, or you really want to do something about it, and that kind of urge should be there to proceed. So I've given you two categories. Those who already have some research experience, please choose the areas which you have had good publications in, because the funding agencies will definitely look into who are you first, why are you applying for this fund, and can I believe you to invest in you so much of funds. It is something like obtaining a bank loan where the bank checks in all your credibilities. It is like that. So for them to believe and take you seriously, you will have to showcase that you are an extraordinary person, or you are a person with very good ideas to really make a very big difference. So either whether you are a beginner or you have recent expertise, you have to have a very good idea. That's very important. And that idea has to be very novel, and it should not have been there earlier. So that is the first step which you have to write down in the paper which you have picked up right now. So at least write down the area which you would like to work on. For example, you've just been listening to bone regenerative materials right now. So the previous speaker was speaking in depth about the bone regenerating materials. For example, I would just quickly pick up hydroxyapatite. Hydroxyapatite, all of us know, the material scientists, we have heard enough of hydroxyapatites, but it's just not that. How many ways this hydroxyapatite can be beautifully changed, how beautifully it can be doped with some other metal, or it can be doped with a growth factor, or it can be doped with many, many enzymes and bioactive compounds or whatever, come what may. Or the HAP itself can be in various forms, the nano form or the mesoporous forms. There are multiple opportunities. So again, taking HAP as an example. So in case if you are a material scientist and for example, any specialty, so we'll quickly, I am basically an oral pathologist, but I don't want to stick on to my specialty. I will say as a from a general perspective. So if you are from conservative dentistry and endodontics, the best department from dentistry to propose a restorative material. Or if you are a surgery background, this surgery background applies for general surgeons as well if they are from medical college. So you can definitely plan in for graft materials, so which with these hydroxyapatite will make. So that should be the idea of interest. So that is the area, the example for area of interest. My expertise is actually more on oral cancer and more on potentially malignant tumors. Additionally, I also have a very strong background on biomaterials. But if you see my profile and my CV, it is more related to salivary biomarkers. So those are the areas which, as a key investigator or as a first author, my most high impact publications are in salivary biomarkers. So obviously, I should take salivary biomarkers more into consideration than other areas according to the agencies. So these are the funding agencies which you have to keep on searching. So it is not always the government agencies. That is why I'm very specifically put this. Please do understand there are multiple sources which are currently available. So you have to quickly note them down, and please do remember, even now, if you go into any of these websites, there will be a call which is open. And right now, I'm sure the female researchers must be knowing, we have a female power grant, SERB power grant open now. So it is closing the coming week. And if you are a female listening to the presentation, you can definitely take that as a challenge and then you can apply. So that is how it goes. So anything, even if you're a male member, there are multiple options which are always open round the clock. There are some grants which can be submitted throughout the year. So the first point which you should have written in the piece of paper right now is, which area are you going to work in? Which area are you going to propose? It might be anything. In case if you don't have a publication and you already have a reasonably good experience, don't worry. You can still propose new ideas depending upon the call. All that you need to do is just keep exploring the options, and that is why this particular slide is there. And the other corporate and international funding agencies also do offer huge funds, but again, do understand there are multiple options. But I have tried my luck with our own Indian company, the Biocon, and that Biocon. Am I audible?
Yes, ma'am.
Yeah, thank you.
Yes, ma'am.
Yeah, thank you. So I've tried my luck with Biocon once and it got rejected. So it is just not easy even with private funding bodies, but still you can definitely try. Biocon is very, very strong and has a exclusive body to take care of these funding agencies for very good thought process and novel ideas which you can definitely propose. So always subscribe into sites like Biotechnica and things like that. So they always keep notifying all the research proposals which are open, and not only the research grants from the government bodies, it also helps you to be aware of the private announcements as well. So the next thing, the first and the second, which we have right now discussed, is the first thing is the core concept, and next is to know the agency, identify the agency, and next is in the core area identified, what are you going to propose? So the idea. So this idea is what made everyone a game changer. So that's very, very important. So that eureka moment would come anytime. So always make a note of it and then think about the feasibility. So it's very, very important. Wait for the eureka moment and then write it. So the idea is actually the key. all of us definitely do have ideas, but many ideas die in the ideation stage itself. Keep thinking about it, and definitely the power of repetitive thinking will definitely make you conspire that particular idea into shape. So when you have an idea, the next thing what you need to understand is think about the study design. Plan on the finer aspects, that is the feasibility. Is it novel? Did anyone not think about it earlier? So do a quick literature search and find out whether it is ethical. This is very important. We will have some very radical ideas. So, but those radical ideas should be suitable to the humankind, and before you take it, it should be ethically conductable. So all that has to be quickly identified, and you can also plan the study design. Which study design will best suit you? So I'm not going into the details of those. So what you do is, there are 3 points which I have highlighted till now. So your paper right in front of you should have the first point, that is your core area. The second point is the agency. The third point is exactly what are you going to talk about. So these are the first 3 points which you have to understand. And the next important thing is the collaboration. Is your idea good enough, or can you alone and your department alone can pull it along, or you need some kind of collaborative research? For example, the BIRAC grant which I am currently having is with collaboration with University of Madras Center for Nanoscience and Nanotechnology, and that particular work cannot be submitted without a proper technical expertise collaborative team. If I would have proposed that idea alone as an oral pathologist, definitely it would have got rejected. But because I took the right collaborator with me along, who have excellent research background, so it became easy. And you already know about the collaborative importance. I can see that PRC is in very active collaboration with Mahatma Gandhi University. That's really very good. That's a way to go. So it is, but again, do understand it is not always compulsory. So you have to first think about whether it is required, and always do understand when ideas merge, when brains, big brains just come into collision, better things come out. There are very, very beautiful ideas which can actually be churned out when you go into a state of collaboration. And again, this is one, I would say, a roadblock, but do understand you will have to think of an idea which is compliant. And again, if there are compliant issues, you can take help from somebody who is experienced in this area, and then try to make it possible. That is, again, it just needs brainstorming. Don't decide on your own that it will not be ethically feasible, nothing like that. Whatever idea you have, discuss it with a peer team who would be really positive about it, or who would really add more value to it. Again, when I'm saying discuss it with your peer team, do understand things are becoming very, very competitive. The moment you bring in such lateral ideas or such beautiful thought process, or your eureka moment, the moment it slips out of your tongue, the moment it slips out of your brain, it would immediately be copied. So, but again, it all depends on whom are you discussing with. Is that team who is listening to are very ethically straight and transparent? Luckily, the team which I've been working with are all very reasonably transparent in all their motives. So it was not difficult, but that is one area which all of us have to be very, very alert, because it's a highly competitive environment right now. So, but you have to have a brainstorming session. So again, I do not have to tell about ethics committee. It has to be ethically compliant. And next is important is the significance of research. So now you have had four listings in your hand. The first one was your core area. The next one was which agency are you going to apply. The third thing was about the idea. And the fourth thing is about the design. And fifth, if it is ethically feasible. So we have completed 5 areas, and then is the most important thing. how novel is your idea is the one thing which you have to quickly do. So every proposal format, the template we say, will definitely have a very beautiful column which says how is the research in the status of research at an international level and status of research at a national level. That has a lot of significance, and the way you summarize the literature in those areas again makes a very big difference, because you can sound extremely confident when you do that part properly. So that is something, just to make it simple, it is just that literature search. But why are they separating national and international? Again, national and international is how many Indian institutions or how many Indian research institutes have done similar work in this area is one question. And how is it at a global area? So that is two questions which has to be answered. Again, when we say national and international, it is not just about national publication, international publication. It's not that. Even Indians who have published in international high impact papers have to be taken into consideration, and that has been conducted in India. That will be checked by the review committee. So the expert committee will go through and they will also check. So the novelty is very, very important. So how novel it is and how different it is, because if it is going to be similar to an existing product or an existing concept, it will be immediately rejected. So always think of some lateral ideas. The same thing should be feasible, the same thing should be ethically correct, and the same thing should have a very, very different thought process, which will make a very big change in the lives of human beings. So that should be the essence of a proposal. So if you can nurture that essence, definitely it would be very, very impressive to the expert committee who is going to scan that proposal form. The next crucial area is budget planning. Most of the times, most of the proposal give you a proper limit. They say plan your budget according to this limit. So when we were writing to DST TIDE, that was my first project in 2015. So that was when during those times we were asked to the upper limit was 50 lakhs, but we were told to restrict our budget to 30 to 35 lakhs. So that is how it went on. And even now I have that fixed thing about restricting it to 30, but not now. We can see a very significant change in the budget allocation right now. It was slow in last 6 months. Now things are better. So even a very interesting oral health call, I'm sure many of you would have applied. The oral health call was 1 crore plus. So that was a very significantly good amount. So whatever it is, so the budget, which proposal you are choosing, and that budget, the upper limit has to be taken. And honestly, how much of amount will be required for your study, how much it is required to conduct your study is again very important, and a very honest budget planning is very important. Do not project it unduly, because it would be immediately understood that you are trying to hike the budget for a lesser valued project. So it has to be very, very truthful, and it should be convincing to the agencies. So again, most of the agencies or most of the proposals come with a project associate area. So the heads is project associate area. Very, very few, like you have a scheme for teaching assistants and research, that is the TARE scheme. Those all do not come with the staff and a header like that. And even there is a scheme called as Young Scientist scheme. So those doesn't support with a project associate. But otherwise, every fund project will have a exclusive project associate in that. And that header, for that header, you can always calculate with the DST's norms of the JRF salaries, and that is what you have to cite. Always refer to the DST bodies for all the funding agencies, so that will be similar to all the funding agencies. So the monthly emolument for a junior research fellow or a senior research fellow is very well planned by DST, and you cannot decide it from the institutional level. So this is one thing which you have to be very, very clear. So that again, if you are trying to reduce it or you're trying to hike it, obviously it will not be very impressive. And what would be the anticipated cost? We have something called as the consumables and the non-consumables, and you can see some word called as contingency grant, which is very clearly for some untoward incident which can happen and which can change the track of the project. So that is some extra additional support which is given, and overhead clearly goes to the institution. So the institution is the one which is giving you the infrastructure, and it is the area where your idea is nurtured or your idea is triggered, and that is the place where you thought of writing of a research grant. So all that, for that sake, the overhead goes there. And literature, of course, I already said, is very, very important. Identify the literature. And the next important and most crucial is the timeline or the Gantt chart. Again, here, if you see, most of the bodies will tell you how many years you can take, whether you're going to take a 1 year, 2 year, or 3 year. That is the common timeline. 3 years is averagely very, very common. There are other proposals which even give a timeline of 5 years. So till date, in my experience with funding bodies, I have seen a max of 5 years. So that is the best timeline which a funding body can give you on a large scale project. Otherwise, the smaller scale projects usually are restricted to 3 years. Some proposals will want a very important pilot run and testing of concept, or which just requires a prototype production and things like that. So that will limit to 1 year. Again, you will have to decide on that. Just like funding, timeline analysis is also very important. For example, if the SERB Power project grant says a minimum of, a max of 3 years, I need not have to stretch my project to 3 years. I can very well give a very practical analysis of my time frame. I might just require 18 months, that is 1.5 years, to complete my project. You can definitely propose on that lines. Nobody is going to say no. But when you are reducing the area of the timeline, then obviously all the budget will actually come down. So that also has to be sensibly cut down in the budget planning as well. So your timeline again is very, very important. So usually what I've done in my proposal writing is, whenever there is a 3-year time given, we usually take a safe of 2.5 years. But sometimes I also stick to exactly what the agency says, and depending upon the proposals which we are proposing. And again, the outcome of the project would actually be really very, very great. It can actually be taken to a phase 1 human trial. So that is what I did with my first grant, which actually came from Department of Science and Technology, New Delhi. So that was a very interesting roller coaster. We proposed for a 3-year, but you know what happened? The DST gave us only for 1 year, and it gave us 28 lakhs for 1 year. So we could quickly establish it was a device for xerostomia. We quickly did the project, and then they asked us, seeing that it was very impressive, we had published our outcome in the British Medical Innovations Journal, and that was very much appreciated by DST, and they immediately agreed to go in for larger clinical trial. That's phase 2 and phase 3 trial. So they again funded 28 lakhs again, additional 28 lakhs for that project, and that came for 2 years. So I had to go through the peer assessment committee with twice, and then everything was done twice. So we have 2 sets of JRF. So the first project was in a different mode, and so it is a very, very different experience which actually the government agencies subject us to. And we also had a very small grant from the Council of Prosthodontics as well. That was like a seed grant, which also happened we supported, applied it along with the prosthodontist whom I was working earlier with. So we applied it to that also, and we had a seed grant in that. So that is how it works. And outcome of the project is what is your end point of your project, whether it can be taken directly to phase 1 trial or whether it would be in which technology readiness level. So that's very important. So this is what is TRLs. So technology readiness level is very important. Anyone who is waiting to apply for write a fund or who has the idea should always do a self-assessment by reading this particular sequence, the TRLs. So whether you are in the idea, do understand idea has a lot of value. I usually respect my students if they come up with an idea. I really encourage them. But I've heard my seniors saying, what is there in an idea? Idea comes to everybody, but I stand to differ because it doesn't come to everyone. So whoever has an idea, you will definitely have to celebrate and cherish. But at the same time, that idea has to be nurtured. It should just not go away just as a matter of talk, or it shouldn't be like that. Immediately, that person should note it down and then keep working on it to make it best, at least do a basic research. So till 3, it is only in the ideation stage. Only when it comes to the prototype stage, you will have people turning and looking at your site. So this again comes with an experience in the device which I had already developed with DST, with that interest and that encouragement which that project gave. I jumped up doing another device. But what happened was that still is in the ideation stage. I have patented it, but I've not done my prototype. So what happened is when I applied it to the Device and Technology Development Board with the DST, again, that was a different call. It's called as BDTD, Device Development and Technology Development Program. I applied and it got rejected mainly because there was no prototype. So that is the problem. So this is when the institution does a lot of support. And again, now I'm working on the prototype with the seed grant available. So once you do it, the seed grant with the seed grant, so to develop a prototype, it will work out anywhere between a minimum of 10,000 to 50,000. That I'm sure all the institutions are willing to offer right now. So that is how it is. And then once you do the prototype, you will have to do a validation. So if it is in the prototype stage itself, it can be, you can apply for really good funding and people will be really convinced. Ideation stage is not enough to impress people upon. So that is where you have to understand. And once it is validated, then you will have to go take it to the commercial system. So right now, 2 of my grants through the DST and BIRAC have gone to TRL 7. They are in TRL 7, state of TRL 7, which we will be taking quickly to the commercialization area. So that is happening. And again, with all this proposal, if the proposal goes and if you are shortlisted, what is, they will be invited for a project approval committee meeting. This meeting is very, very critical. You will have a panel of 12 to 15 members sitting there to rip you apart into pieces. That's not an exaggeration. But again, what I would like you to feel is, it is your project, it is your brainchild. You will have to feel proud about it. Just walk with your heads high, chin up, and propose it with full confidence. Only then that confidence would be conveyed to the members. The moment you sound lesser confident, they will never want to give the fund with you. So you send a proposal and then comes the peer committee's intervention, and that meeting, getting selected to PAC meeting itself earlier times when I got my DST was like approved. It was like that 5 years back. Recently we had a very bad experience. I had an Indo-German call worth around 3 crores with a very top researcher in Germany. That was again a biomaterial research call. It came to PAC meeting, but we were surprised to receive a rejection notice after the PAC meeting. So it can be otherwise. So it's always fingers crossed till you get the sanction order. But whatever it is, I've never lost hope. And the moment you have this sanction order in front of you, it feels just great. It feels so, you feel so happy. you feel so convinced, and nothing can beat receiving this particular order in your mailbox. I cannot tell you how it feels, but at the same time, there have been multiple times of rejection letters as well. But anyway, that two things which actually keeps me going, and the other smaller agencies, those are actually gives us a lot of what to say, knowledge, and the way we grow every day, and nothing leaves you without teaching you a lesson. So even rejections, you can actually welcome it with a warm heart and then make it better and apply again. And then once you get the sanctioned report, you do work on it, and then you have to do a completion report. So this is how a research grant goes. So first thing is identify your core area, identify your the idea behind, identify the agent, and then start writing a very good draft. All my colleagues and my seniors always encourage to get the proposal ready at least a month before. But you'll be surprised, I'm always a person who would do it at the last minute, and that is something which is not a very good thing. So it is better to be ready a month before, and it should be 3 weeks before you submit. 3 weeks before the deadline, you should be able to submit. By doing that, you will really do a thorough work, and you would have done a very intense deliberation among your peers, and it would really make a very big change. So once you do that, you actually, if you're offered a grant, you set it up, and then you spend it, report it, and then do grant closure. And believe me, what it has done to me is actually very important. So after I started receiving grants from the year 2015, my research profile totally changed. Earlier, before that, I was the same, with the same urge and same energy levels, but my research graph was not going in peaks. But after the grant, my publications, everything started going into peak areas because of the availability of fund. So that is what is the importance of the fund, and that makes a very big difference in your research career. So if you are a person who is very passionate about research, writing a grant is important. And again, writing a grant is not very, very easy. You will have to plan it well before in time, and always have a model proposal. Take the help of your seniors, and that makes a very big difference. So have a model proposal and then use that up. So lucky for me, I was having a lot of model proposals in time when I wrote my first proposal. So that is when the first proposal which I wrote to DST type got clicked, and then there was no looking back. So as you keep investing, you know that there is a lot of things happening, but it's not always easy. There have been multiple roadblocks in the way, and I've only been telling my success stories. There have been multiple rejections as well, but always do understand, you should always remember to stay strong and never give up. Every time when I receive a rejection letter, either from the publication agency, or from the journal, or from the agency, the funding agency, it really pains. Okay, it really pains and it hurts. An immediate reaction, a normal human typical reaction even comes to me, and I'll think, I will never again write a fund. But that lasts only for some time, and again I come back and then start applying again and again. So that is how it goes, and it's real fun because you should keep on doing. And one of my collaborators from National Center of Nanoscience of Technology, University of Madras, Balakumar sir keeps telling me always, you will have to fight like Ghajini Mohammed, who won over India, northern part of India, by not first attempt. He had to keep on trying. So that is the power of Ghajini. So it is just that repetitive trying and repetitive attempts which actually will make you relish your dreams, and you should never, never give up. And even if the proposal doesn't work, over the years, I've learned that that proposal itself can be slowly done in a smaller way, and we can do a wonderful publication with that. Because don't allow those ideas to just go away. Take some, make it smaller, into a smaller version, and at least do a publication with that. So never ever any effort done with a complete dedicated heart and mind. And if you give your full soul and energy to it, it will never ever go for a waste. You can make use of it in any ways possible. So start writing a project today itself, because there are multiple calls which are open, and I would like to acknowledge all these heads. I have a few names included from other institutions than which I work currently, mainly because these are the people who introduced me to the world of research grants, and being with them taught a lot to me. And that is how you should really learn from people who had really won grants and who have the experience of writing multiple grants, and you can really make know things better. Yeah, so I'm open for questions.
So thank you, ma'am, for that captivating lecture. And your years of experience in this field is evident when we listen to your presentation. So like ma'am said, any questions, anybody? Yeah, if not, I think I'll just say that what ma'am said, when you get that first sanction letter, you feel so proud of yourself. It may be after 10 rejections, but that one letter, you know, it's worth it.
All the differences.
Yeah, so please keep trying. Those who are applying, please don't give up hope if you have not yet got there, but you'll definitely be successful. You have to just keep trying.
Yeah.
Yeah. So thank you so much, ma'am.
Thank you again. Thank you.
Thank you, Dr. Ramya, for the session, and thank you, Dr. Shirley Anthony, for the moderation. Now it's time for conclusion of the BioRadiance 2022, and I would like to invite Dr. Nebu George Thomas, the organizing secretary, for the concluding remarks and word of thanks.
So for the past 2 days, we had a wonderful sessions on translation research. So it is indeed a great privilege to have the speakers from the various fields. Those who are working in the field of epidemiology, community medicine, material scientists, clinician, and also from the academician, including the reputed vice chancellor of MG University, is one of the renowned scientists in the field of material science, has joined us today. So I would like to thank all the speakers for sparing the valuable time with us. And also I would like to thank the delegates, those who are representing the various field of medicines like dentistry, general medicine, nanoscience, materials. So I would like to thank all the delegates and also the students from the various fields like PhD, undergraduate students, postgraduate students of the field of medical, dental, and basic science. So I would like to thank, and I would like to announce that this should not be the last program, but it will be a first program regarding the translation research, and shortly we will come in with the offline meeting. And also I would like to thank the management of the Pushperi Medical Society, especially the Director and Head of Research, Dr. Father Matthew Majanjal, and also the Director of Pushperi Dental College, Reverend Father A.B. Vadakundala, for their valuable support. Without their support, we will not be able to organize a meeting like this. And also I would like to thank all the organizers and the members of the scientific committee, mainly the Dr. Saumya, Dr. Sharif, Dr. Meher, Dr. Sunu, and also the moderators of the various HODs of the Pushperi College of Dental Science for supporting us. Thank you. So we have shared a feedback form, so you can fill the feedback form and you can email to the same email ID.